Let's go ahead and get started. Today is Thursday, April 22nd, 2021. We are continuing our discussion of the nervous system. We are almost done with our nervous tissue. We've actually talked about most of the things that we were going to talk about today already. We're going to add a little vocabulary to some things and add a few tiny things to it. But most of the lecture part, at least at the beginning, is a lot of kind of review of concepts we've already worked with. We're just rethinking them. And then what we really need to spend a lot of time do is catching up on our brain anatomy. If we can get through that today, then we will move on to the protections of the nervous system and even from there on to the spinal cord. So that will be our game plan for today. The rest of the schedule is here on the board. We have on Tuesday next week, your PhysioX exercise 3, which has 9 activities to do, so 9 assignments on that. I would also strongly encourage you to print out the cranial nerve handout. We are going to be working on that together, so you don't have to fill it out ahead of time. But as I have warned you, depending on how things are going, and because we are already a little bit behind where I want to be right now, this could be as much as 25%. Oops, that's not a 5. 25% of your lab exams. So it is going to be important that we understand these concepts, and either having that handout or something, some kind of graph setup like that to help you to go through that information, I think is something that will definitely be helpful. Thursday next week, your 15-point nervous, apparently I don't know how to spell nervous, review is due. And remember, that is graded for correctness, and it really emphasizes that relationship between structure, function, and location. Those things that we have talked about and will continue to talk about even more as we move on today and into the spinal cord. Unit 14 review is due, and then there's a reflex lab due on Thursday the 6th. Then on the 11th, the following Tuesday, we have our lab and lecture exam, same as it ever was. You know exactly what to expect from that. As someone pointed out, on Thursday, the 13th, we do not have class. That is considered part of finals week, so we will not have class on that day. So we do not meet again till the following Tuesday, the 18th, where you will have your cumulative final exam. And again, it covers every single thing we have covered in this class with 100 multiple choice questions. So you don't have to pull the information out of the ether. You just have to be able to recognize the correct answer. And it absolutely positively counts as a grade on its own. But remember, you also have the opportunity to replace your lowest lecture score with it. So for some of you, great question. So for some of you who had an exam where you struggled, this could be a really good way to boost. Arthur, your grades have been tabulated for the last lab and lecture exam. They have been posted, so you should be able to see your grades. You should have gotten a notification from Canvas about that. And then today after class, I will release it like I do for 24 hours so that you'll have the opportunity to be able to do that. So absolutely, if you didn't get that notification, if you don't have notifications set up in Canvas, then yeah, you might not have gotten that right away. But we will... that will be released today, so you'll have the opportunity to look at it, and the grades are already posted. I just don't like to post the exams before class because then people are busy looking at that and not listening to lecture, and I want you listening to me. All righty. Any questions on any of that? All right, excellent. That's the game plan, with the potential exception of there probably being one more daily quiz somewhere along the way. So somewhere on one of those days, we'll have something like that. So again, always important to be here and be on time. All right, excellent. So last call. Any questions on this before we move forward? All righty then. We left off last class, and let's just do this here. We have talked about the anatomy of a neuron. We have talked about how on the cell body and the dendrites, we can receive signals. Those signals are typically either stretch or chemical in nature, where they open up chemically gated channels, or mechanoreceptors producing those graded potentials. Those graded potentials spread to our one single axon, where if that graded potential is big enough ataraxon hillock, we can produce an action potential. So we've talked about both graded and action potentials, and propagated that action potential down the axon, both with and without myelin. So as far as the function of the neuron that we've talked about, the only thing we haven't talked about now, but we actually already talked about before, is what happens when it communicates to its target cell, or more specifically, how it communicates to the target cell. And while we may not have always used the name, we do know the name for the general specialized region of communication. What is that name for that specialized region of communication between a neuron and its target cell?
Synaptic cleft?
Well, that is the space in between, absolutely, and that is a part of it. But what is the general term that we give? There we go, the synapse. The general term we give for it is absolutely indeed a synapse. Is that general term for that, absolutely. So, and luckily for us, we know how synapses work. As I mentioned, what we may not have said from a vocabulary standpoint is that there are actually two types of synapses. All right, let's go back to the whiteboard for a second. Way back at the beginning of 430, we talked about how cells have cell junctions. And one of those cell junctions, I'll cheat here, involved a special protein called connexons, which were transmembrane proteins that embedded through the plasma membrane of one cell and connected to those in the plasma membrane of the other cell. And in doing such, they formed a fluid-filled pore that would allow water and stuff dissolved in that water to move directly from one cell to another, right? What did we call this type of cell junction?
Was it a gap junction?
It was a gap junction, absolutely. This cell junction was a gap junction, right? Formed by connexons. Excellent. This is not necessarily a new piece of information, but what we can say about this is that there is another term we can use to describe this. Essentially, this physical connection between the two cells is also what we can call an electrical connection, or more specifically, an electrical synapse. So what we're saying here is that there are actually two types of synapses, and you've already studied both of them. Now, as you know, typically the propagation of an action potential comes from a large wave of sodium entering into the cell, and it produces an action potential. Well, if that action potential were able to flow through a gap junction to the next cell, then that next cell would basically, that action potential would basically spread from one cell to the next. And that's essentially what an electrical synapse does. It allows the current of ions to flow directly into the next cell. Basically, these cells are essentially hardwired together, like the wires in your house that we talked about. You flick on the light switch and the light turns on. You flick the light switch off and the light turns off. And no matter how many times you flip that switch 100 times, that light's going to come on 100 times. You're never going to flip that switch and get your garbage disposal to go on, or your garage door to open, or the back door to swing open, or something else. Excuse me. It's not going to happen as a result of that. These things are hardwired together. There are some advantages to that. Obviously, they work together as one. True. Yep. The cells work together as one. All right, with this. Also, while I had the cell on the left firing the action potential and spreading it to the right, it can work just as easily the other way as well. If the cell over here on the right fires the action potential first, it can spread it to the left. So the cells work together as one. It allows two-way communication. Between them. And oops. Does it take a long period of time from sodium to move just from one side of a cell to another through a small hole? No, of course not. It's a fast process. All right, so again, this is our electrical synapse. One of our two types of synapses. Of course, the other type of synapse that we've been talking about, like we saw at the neuromuscular junction, involves the synaptic end bulb of one cell communicating with, in the case of our muscle cell, the motor end plate. But basically we have here, if we're going to just go with a generic synapse here, we have the presynaptic cell. As we know, that presynaptic cell is going to have some specializations to it. It is going to contain vesicles, and those vesicles have what inside of them? True, acetylcholine is an example of what it could have inside of it, but remember, it doesn't have to be acetylcholine. What's the generic term we would use for that?
Neurotransmitter.
Excellent. Protein is right also, but a little too generic. A neurotransmitter. We have vesicles containing neurotransmitters that are going to produce our communication, and we need some way of controlling those. How do we control the release of the neurotransmitter?
With calcium.
With what?
Calcium.
Calcium, excellent. We let calcium in, and that calcium has to come in what?
A voltage gated calcium channel.
Excellent. So our other specialization here is a voltage gated calcium channel. Excellent. We also then have our target cell, which of course we also would call our presynaptic cell, post. The wrong size. Nope, that looks better. We have our target cell, our postsynaptic cell, and what is going to be the primary specialization of our presynaptic cell?
Chemically gated channels.
Yeah, chemically gated channels. Excellent. We are going to have some type of chemically gated channels that will allow ions into or out of the cell, causing some type of change. Here is our synapse. We've been calling it a synapse, but essentially what this is what we call a chemical synapse. With this chemical synapse, notice instead of just letting the ions flow straight through, wow. It allows, let's say it this way, the release of a chemical signal opens ion channels in the postsynaptic cell. And that ion flow produces our current. Notice here, the current just flows straight through. Over here, but here we basically use one current to release the chemical, and then that chemical produces a second current. Now, notice one of the big differences between these two is while this one here can go in both directions, it can be a two-way communication. Does this allow for two-way communication?
No.
No.
No, this is one-way communication only. And instead of just having sodium flow from one cell to the next, as we know, because we talked about it in the last section, we have a action potential coming down the neuron, which depolarizes our synaptic end bulb to threshold, opening those voltage-gated calcium channels, allowing calcium into the cell. That calcium triggers the exocytosis of the neurotransmitter, which into the synaptic cleft, which spreads across the synaptic cleft, binding to the chemically gated channels on the other side, opening up those channels, allowing ions to flow. And if that signal is big enough, we get an action potential in the second cell. Is that anywhere near as quick as sodium floating from one cell to the next?
No.
No, it's a relatively slow process. We'll put slow in quotes. Again, most of you haven't thought of it in this way, but you're absolutely aware of it, because one of the fun things you can do on the interwebs, or there's lots of other places where you can do it as well, is you can do reaction time stuff, right? Where there are, like, for instance, websites that you can go to that have a red light, and as soon as the light turns green, you slap the space bar, and it tells you how quickly you have reacted to that stimulus, right? And there's a delay, 200, 600 milliseconds, something like that on the delay. We'll get to that in a second. We will absolutely talk about that in a second. So it is, so again, when I say relatively slow talk, we're talking about fractions of a second, but it could be 200, it could be, 600, 800 milliseconds in that delay. And a lot of that delay that you're seeing in that reaction time is due to this time that it takes for that chemical signal to stimulate the second cell. So this is a slow process. So if this process is so much slower, let's actually answer Yulia's question. So this one only goes one way, this one's slower. So which of these two types of synapses, either the chemical synapse or the electrical synapse, do you think you see the most of in the nervous system? Both of these are found in the nervous system, but which one do you think there's more of in the nervous system?
Electrical.
Electrical? Absolutely, because that would make a tremendous amount of sense. Oh, wait, someone says chemical, which would also make a tremendous amount of sense. So who's right? We've heard both now, electrical and chemical.
Electrical.
Turns out that absolutely positively most synapses in the nervous tissue are chemical. Why? If the electrical's faster, if the electrical's two-way, why are most of the neurons in our nervous system chemical in nature?
Wouldn't it be because the nervous system controls all the slow things too, like breathing, temperature, but the fast electrical synapses would control more like muscle movements and stuff?
Well, but let's think about that. We already learned how to control a muscle. What kind of synapse was found at our muscle? Was it chemical or was it electrical?
Chemical.
It was chemical. The neuromuscular junction is a chemical synapse. The real answer comes back to that last part of what I said when describing communication at that junction, right? I said, if the signal is big enough. The implication there is that it's not always going to occur, right? When that light switch turns on, the light goes on, right? No matter how many times I flip that switch, the light is going to go on. You have a reflex in your knee that you don't voluntarily control, that when the doctor hits your knee with a hammer, what happens?
You extend your knee.
Yeah, your leg kicks out. Absolutely. You extend your knee. Excellent. Right. And then if he does it again, you're going to extend your knee. And then if he does it again, you're going to extend the knee. And then he's going to put a sleeping newborn baby on this right in front of your foot. And then he's going to hit you in the knee. And when he hits you in the knee with that sleeping newborn baby right in front of your foot, does your leg kick out?
Yeah.
No.
No.
Or no.
Well, I guess it depends on what kind of person you are. All right. A sleeping tiger. He puts a sleeping tiger in front of you. Are you going to kick your knee out then? No. The
No.
advantage of a chemical synapse is that this type of synapse, the response can be modified. I've used the example earlier in this class, right? If your nervous system was hardwired using electrical synapses, right? Then of course, when I step into the room, open the door where you're at, holding my red pen in my hand, look you in the eye and say, I'm going to kill you. And a bear with an axe opens your door, looks you in the eye and says, I'm going to kill you. Are you going to respond to those two stimuli the same way?
Yeah. Or
No.
Trust me, I'm not, while I may seem threatening in this class, me with a red pen is not anywhere near as scary as a bear with an axe. Right? So absolutely, you're not going to respond to those things the same way. So the advantage of a chemical synapse, and the reason why most of the synapses in the nervous system are chemical, is because the response can be modified. Sometimes we can get an action potential, sometimes we won't. We can change the behavior. And that's really the key. We can change the behavior. Even if it's something that is, like I said, like a reflex. You touch something hot and you put your hand away from it. Right? However, if you're trying to walk across coals, right, then those hot coals, then when you step on it, you don't necessarily pull away. You control that reflex. Right? When that sleeping tiger is in front of your foot, you're not going to swing your foot out when he hits you in the knee with the hammer. We have the ability to modify our responses, and that's really the key to a chemical synapse. So again, electrical synapse. Go ahead.
So what muscles or like what uses the electrical synapse? Can you give us examples of that?
Well, so for instance, cardiac muscle, we talked about, has gap junctions because we want the heart to work together as a single unit in its contracting. Remember, we talked about how single unit smooth muscle, the whole layer of the wall of the stomach, for instance, works together in the churning of that. So we see it in areas like that. There are a couple areas in the nervous tissue that use it, but very, very few. Most of the connections in our nervous system involve chemical synapses because we want to be able to modify our behavior. So I won't say there are none, because that's not true, but most are chemical. So again, electrical synapses use those gap junctions. They're fast. They allow two-way transmission. They allow the cells to work together as one, but they're typically not found in the nervous system. Instead, they're found in smooth muscle and cardiac muscle and places like that, because again, those are places where it's okay for things to be hardwired together. Right? When we want to make a decision on our behavior, we want to use chemical synapses. Here, the two cells don't touch. There's that synaptic cleft in between. Our presynaptic cell releases neurotransmitter. Right? It takes time for this neurotransmitter to be released, time for it to go across the synapse. Right? It only allows that one-way transmission, but we can modify the activity. We can modify the influence. And how it affects the cell can be very, very different as well. It can be both excitatory or inhibitory. Again, if we think back to what we were just talking about, let's go ahead and draw two cells here. Here's one neuron. Oh, someone just wrote a paragraph. All right, hold on. Give me a second here. Here is a second, and then let's put my target cells. Let's talk during the break. All right. So both of these are capable of releasing a neurotransmitter. Ding, 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 ding. However, if one of those neurotransmitters, for instance, were to open a sodium channel, what would happen?
Sodium would enter the cell.
Excellent. Sodium would enter the cell. And what would happen to the cell when sodium entered it?
It would depolarize.
It would depolarize, right? It would get more positive. Excellent. But what if instead that neurotransmitter opened up a potassium channel? What would happen then?
Potassium.
Leave the cell.
Potassium leaves the cell. And what effect would that have on the cell?
Becomes more negative.
And what is the term we use for that?
Polarizes.
Hyperpolarizer. Remember, if it goes below rest, it's hyperpolarizes. Excellent. So not only can we modify the behavior, but notice by opening up different types of channels, we can have different types of effects. Now, these two types of effects, as we mentioned, depolarize and hyperpolarize, right? Those are talking about their relationship to, actually like that. Let's write this here. When we talk about depolarize and hyperpolarize, we talk about the relationship to the resting membrane potential. Now, I have one other quick question for you before we get to this next point. What do you think the most common target for a neuron is?
Sodium channels.
True. Okay, but I mean, what kind of cell? What's the most common target cell for a neuron? What do most neurons talk to?
Muscles.
Muscles is a good guess, but we have, not muscles. It's a good guess, but it's not muscles.
Other neurons?
Other neurons, absolutely. The most common target for a neuron is a second neuron. So if you think about it, the goal of stimulating a neuron is to try to get it to fire an action potential. So when we talk about depolarize and hyperpolarize, we talk about the relationship with the resting membrane potential, but we can also use these terms excitatory and inhibitory. When we use the terms excitatory and inhibitory, we are talking about the relationship with that magical point you have to reach to fire an action potential. And what is that maximal point we need to reach to, or really it's, I guess, the minimal point we have to reach to activate an action potential?
Threshold.
-60.
Threshold, exactly. Which, of course, you guys are correct, is -60 millivolts. So when we talk about the relationship to the threshold, right, that tells us whether or not we get an action potential. We talk about them as being excitatory or inhibitory. Now, let's take a look at this. Here, of course, is our chemical synapse. You had to know how this thing worked in the last class. I just reminded you of it again. So I'm not going to go over it a third time because hopefully at this point we've got that. But what I do want to talk about is what happens when that neurotransmitter binds to our postsynaptic cell. So again, we have our first neuron with its axon, with its action potential. And now we know that the most common target is a second neuron with an axon and an axon terminal. And I guess we need to give these guys their squiggly hair, their dendrites. Because someone pointed out that dendrites are what most neurons are going to be communicating with. The axon of one communicates to the neuron. The axon of one communicates with the dendrite of another. And their goal, like we talked about, is to open up that channel, a chemically gated channel, get ions to flow in and out, right? This is what we called before a graded potential. When we talk about these graded potentials, right, there we go. These are occurring in our postsynaptic cell. We use the term target cell, so it could be a target potential if you want it as well. But I think presynaptic and post cell and postsynaptic cell are useful terms. So when we produce a graded potential in the postsynaptic cell, it is a postsynaptic potential. Again, this isn't anything we don't already know. We're just adding a label to it. And as we talked about, we can relate this postsynaptic potential to whether or not it's more likely to produce an action potential. So it can be excitatory when it depolarizes, or it can be inhibitory when it hyperpolarizes. Again, no new information there. An excitatory postsynaptic potential, which is a mouthful, but the good news is once you've written it out once, you can abbreviate it EPSP. Excitatory postsynaptic potential is one that causes depolarization. Because when you depolarize the cell, you get the cell closer to threshold. Now, notice the key word in that sentence. I said closer to threshold. Did I say it had to actually get it to threshold?
No.
No, it does not actually have to reach threshold to be considered excitatory. All it has to do is get it closer. It has to make it more likely. And let's actually emphasize that. Make it more likely to reach threshold. If it makes it more likely to reach threshold, then we consider it excitatory. And based on what we've talked about, what type of channels, or let's say it this way, what ion are we going to most likely use with this type of excitatory synapse?
Sodium.
Yeah. Absolutely sodium. Again, you guys have mentioned it before, so I'll remind you of it again. Technically, calcium would work the same way, but remember, calcium makes cells do wonky things. So most of the time, we're not going to use calcium because calcium causes problems because it causes wonky things to happen. So most of the time, we are going to use sodium. Excellent. So again, an excitatory postsynaptic potential makes it more likely to reach threshold. It doesn't actually have to get there, and it typically involves sodium channels. Conversely, an inhibitory postsynaptic potential, which again, after you've written out once, you can then abbreviate IPSP. It's an inhibitory postsynaptic potential. This one moves the membrane potential further from threshold. Makes it less likely to reach threshold. Again, it doesn't actually have to stop it from reaching threshold. The typical neuron gets something on the order of 10,000 inputs to it. So if the neuron we're talking about here, our target cell, gets 1 inhibitory and 9,999 excitatory, do you think it's going to fire an action potential?
Yes.
Yes. But this one that made it a little less likely is still going to be considered inhibitory. So it makes it less likely to reach threshold, but it doesn't have to stop it. Excellent. And what type of ion channels would we most likely use for inhibitory postsynaptic potential?
Potassium.
Excellent. And notice I did say channels this time. So potassium, and what would the other one be?
Chloride.
Yeah, chloride ions. Absolutely. Because remember, those chloride atoms, they're negative and they want to come in. Potassium's positive and it wants to go out. So both of that will make the cell more negative. If it's more negative, if it's hyperpolarized, it is inhibitory because it is moving further away from threshold. So again, makes the cell less likely to reach threshold and typically involves chloride or potassium channels. Excellent. So again, as I mentioned, notice so far the things we've talked about, there's really not a lot of new information. Really, all we've done more is added vocabulary to what we already knew. Things like inhibitory postsynaptic potential, things like chemical synapse or electrical synapse. We really haven't talked any new concepts yet, but we have kind of added a few layers on and added some vocabulary to this. So don't let it confuse you too much because absolutely these are all things that we have talked about already. All right, the last thing we need to talk about then is about these. As I mentioned, a typical neuron gets somewhere between 1,000 to 10,000 inputs to it. It has 10,000 synapses. So one neuron has the potential to receive input from over 10,000 different neurons carrying information in. And really, neurons are bean counters. They're accountants, right? What does an accountant do?
Counts thing.
Yeah, absolutely. It counts things, right? Mostly that accountant counts like money. If last month, like me, right? Because yeah, I've got that kind of scratch because I'm a college professor. I spent $1 million on bills. My kids really like to shop, right? Is that a good thing or a bad thing?
It's a bad thing for your wallet.
Well, but it depends. What if because I'm TikTok famous, I made 2 million last month?
Make it rain. Make it rain.
There you go. If I made 2 million last month, then is spending 1 million a bad thing?
No.
No. If I made $100 last month and I spent $1 million, then that's going to be a bad thing, right? So notice it's not about the absolute numbers. It's really about the differences between them. Right? As we know, and let's think about this with simple math, a graded potential, remember, as we said, on average is about 2 millivolts. So if I just produce one excitatory graded potential in a target neuron, is it likely to fire an action potential?
No.
No. How big of a
No.
change do I need to reach threshold?
900.
10 millivolts.
I need 10 millivolts. So I would basically need 5 excitatory postsynaptic potentials. But I need to clear 5. Like I said, if this cell gets 5,000 excitatory inputs to it, and then 5,000 inhibitory inputs, it's receiving a tremendous amount of information. And what is this neuron going to do in response to it?
Nothing.
Nothing. Because the 5,000 positive are going to cancel out the 5,000 negative. All right, they're accountants. If, on the other hand, I receive 7,000 excitatory and only 3,000 inhibitory, is this neuron likely going to fire an action potential?
Yes.
Yes. And while 4,000 excitatory inputs sound like an awful lot, If the cell receives 6,000 inhibitory, is it going to fire an action potential?
No.
No.
No. And so that's really all neurons are. In our central nervous system, when we talk about processing information and making decisions and doing all those things, at its biological core, this is all those neurons are doing. They're counting beans. They're counting positive inputs and negative inputs and deciding whether they're going to fire an action potential or not. So to get a cell to fire an action potential, you've got to basically fill its bucket. Or your textbook, I'll be honest, I haven't read this part in your current textbook. I hope it kept it because I really thought it was an excellent analogy. The analogy that your textbook used to use, and hopefully it still does, is they think of threshold as like being the top of a bathtub. You have to fill a bathtub with hot water to its top. If you do that, you reach threshold and you can fire an action potential. And let's say it takes 10 buckets of water. To fill that bathtub. Excellent. One way I could do that is I could, because my bathtub, of course, is up here on the 4th floor of my house, because again, I have that kind of stretch. But the only working faucet in the house is in the basement. So I have to go down to the basement, fill up my bucket, come up the 4th flight of stairs, dump it in the bathtub. Then back down to the basement, back up here, dump it, right? And if I do that 10 times, I can fill my bathtub, right? Because it takes 10 buckets of
Yes.
water to fill the
Yes.
bathtub? Yeah, absolutely. But here's the problem. The problem is I've lost my stopper for the drain. So I dump that first bucket into it. I go down the 4 flights of stairs to the basement. I fill my bucket. I come all the way back up the 4 flights of stairs to my bathtub. And what's happened in that time?
Some or
Already
all of your
drained.
water is drained out.
Yeah, and my guess is if it's 4 flights of stairs, that it's going to have all drained out, right? And I'm never going to be able to fill it that way. So what I have to do is I have to get 10 buckets of water in here very close together. And not surprisingly, there are 2 ways they can be close together. They can be close together in time, or close in space. Yep. Although in fairness, in California, having a basement, right, that is an oddity. So, of course, it's over an Indian burial ground, so there's a whole issue with that as well. All right. So we got to get these 10 buckets of water into my bathtub, either close together in time or close together in space. So not surprisingly, I'm sorry?
Sorry, what does close in space mean? Sorry.
Well, that's what I'm getting to right now. So
Okay.
we call these 2 types temporal summation, and then the other one is spatial summation. You're right. I think, again, calling it close in space doesn't make as much sense, but I promise you I'll explain it. Option 1. Right? I could do this a couple of ways. Yes, my faucet is down in the basement, but maybe I run a hose from that all the way up here to the 4th floor. So while I'm sitting here, I can be filling buckets and dumping buckets right away. So fill, dump, fill, dump, fill, dump, fill, dump, fill, dump. Me by myself can fire fast enough because we know neurons can fire super fast. They have a very, very brief refractory period, matter of seconds. So one neuron by itself fires frequently enough to produce enough graded potentials to reach threshold. Right? So me standing there by myself, rapidly dumping my 10 buckets, I can get it done close in time. All right. But how fun is a bath by itself? So instead, I decide to get 9 of my closest friends. And 9 of my closest friends all stand around the bathtub and we all hold a bucket of water. And at the same time, all 9 of us being in the same general location, all 10 of us dump all of our buckets into that tub at the same time. And if me and 9 of my closest friends all dump our buckets at the same time, can we fill that bathtub up to the top?
Yes.
And that is what spatial summation is. Spatial summation is multiple neurons with synapses in the same location. So that's why it's called spatial summation. So multiple neurons with axons in the same location fire together. And that produces a big enough signal to reach threshold. All right, so that's how our neurons work. Either one neuron can fire very rapidly to make a big enough signal, or many neurons can work together to make a big enough signal. And if we can fire fast enough or work together with enough cells, we can reach threshold and we can fire an action potential. All right, let's look at, instead of my pretty picture here of the bathtub, again, we have this bean counting. 1 excitatory postsynaptic potential is canceled out by 1 inhibitory. We did that already. And as I mentioned, there are 2 ways that we can add graded potentials together to reach threshold: spatial summation and temporal summation. And here's the pretty pictures. Notice here, 1 neuron working by itself fires, and while that graded potential is there, it fires a second time and then a third time, and it adds those together, close together in time, to reach threshold at the trigger zone and produce the action potential. Or spatial summation, right? Me and my closest friend all dumping our buckets at the same time, producing the 2 multiple neurons, producing graded potentials at the same time, add up to a big enough signal to reach threshold. All right. We talked about how in our muscular system, 1 neural action potential meant 1 muscle action potential meant 1 twitch. But our control of our muscles is far simpler than the communication and the processing that we want taking place in our nervous system. So typically in our nervous system, 1 neural action potential in 1 cell doesn't produce 1 neural action potential in the next cell. There are some that work that way, and we'll see that in the autonomic nervous system, but not a lot, because we want to be able to modify, we want to be able to process, we want to count beans, get information from lots of locations, and make a decision based on that of what we're going to do. All right, questions on that? All right, excellent. Last thing we need to talk about for this portion of the lecture is, again, we keep talking about neurotransmitters, and of course we know our most common neurotransmitter in the peripheral nervous system, which is acetylcholine. But as I also mentioned, over 100 neurotransmitters have been identified in the human body alone. Other organisms have even different types of neurotransmitters. Now, when we talk about neurotransmitters, typically we refer to them as excitatory or inhibitory. By, of course, because of the relationship they have with the target cell. But things get tricky with that. After all, as we know, I have a somatic motor neuron, and that somatic motor neuron communicates with these long cylinder cells that have these striations on them. This cell too long. And pushed up against the outer surface of its plasma membrane are multiple nuclei. What have I just drawn here?
Muscle fiber.
Yeah, but what
Skeletal
kind?
muscle fiber. Skeletal.
Yeah, skeletal muscle fiber or cell, right? Either of those are acceptable. Excellent. Now, as we know, what neurotransmitter is released here by this somatic motor neuron?
Acetylcholine.
Acetylcholine.
Excellent. Releases acetylcholine. And of course, as we know, what kind of effect does it have? Is it going to be excitatory or inhibitory?
Excitatory.
Excitatory effect. Excellent. Excellent. That's all perfect. So of course, that means acetylcholine is an excitatory neurotransmitter. Except for one minor problem. It turns out some of our autonomic motor neurons, specifically the ones of the parasympathetic nervous system, as we know, being part of the autonomic nervous system, they talk to things like some branching cells. These branching cells, as it turns out, also tend to have stripes on them. And one big centrally located nucleus. And of course, what type of tissue is this here?
Cardiac. Muscle tissue.
There you go. The cardiac muscle fiber. And what's interesting is that this parasympathetic nervous system neuron also releases acetylcholine. And so does that mean that it is going to be excitatory on our cardiac muscle? If these were ancient times and we were on campus, one of the things you would do is you'd be catching frogs and pithing those frogs in our 431 class when we were learning about the nervous system. For those of you who don't know what pithing is, you basically take a very large bore a syringe needle and basically hold the frog and take that needle at the base of his spinal cord and run the needle down his spinal cord, completely destroying his spinal cord, keeping him alive, but paralyzing him completely. This allows you to pin him down without him fighting, skin him, open him up, crack open his rib cage, and watch his heart beat while he still lives, but can't feel it. And you can actually take a little acetylcholine then and sprinkle it onto that heart. And is the heart going to beat faster when you put acetylcholine on it?
No.
No, it
No.
turns out the heart beats slower. So in this case, on cardiac muscle, that exact same neurotransmitter, the acetylcholine, actually has an inhibitory effect. How can that be? How can a neurotransmitter have both an excitatory and an inhibitory effect? Doesn't it have to be one or the other? How can it do two different things? What do you guys think?
Maybe it depends on which channels it opens.
Exactly. While we typically refer to neurotransmitters as being excitatory and inhibitory, their effect is truly determined by the receptor. As of course you know, because we have already talked about it, over here on our skeletal muscle, that acetylcholine is opening up cation channels. And for that cation channels, as we know, it is primarily sodium that enters the cell. And that is why we get a positive or excitatory effect. Well, it turns out over here in our heart, we have a completely different type of channel. And this channel actually allows chloride in. This is a chloride channel. And of course, when chloride comes in, it makes the cell more negative. So it has an inhibitory effect. So acetylcholine is a key that unlocks two different types of doors. So you'll notice as we talk about the neurotransmitters, we talk about them as being excitatory or inhibitory, but really it's their receptor that determines their effect. Okay, let's take a look at some examples. As we have already talked about, there you go. Oops, here we go. So it can be excitatory or inhibitory depending on the postsynaptic effect. Also, how strong an effect can be determined by a couple of factors as well. How many action potentials you produce in the neuron can determine how much a neurotransmitter is released. More action potentials. So let's put that up here. More APs equals more neurotransmitter. However, it doesn't just take more action potentials to release more neurotransmitter. We can put more neurotransmitter in each bucket. Of course, we don't use buckets, we use vesicles. But if we put, there we go. If we put more neurotransmitter in each vesicle, then that will work as well. We can also release more vesicles. Release more vesicles when we do that. And notice, like we talked about, and some of you had as a essay question on this last exam, right? If we, for instance, one of the possible essay questions was what happened if you removed acetylcholine esterase from the synaptic cleft. And as I'm sure you all know, or if we're figured out since then, that if you remove that, you take away one, but not all of the ways we get acetylcholine out of the synaptic cleft. It can still diffuse out, it can still be endocytosed, but by removing acetylcholine esterase from the synaptic cleft, the acetylcholine stays in the synaptic cleft for longer. So it has a longer effect on the muscle cell. So typically the muscle cell has a longer contraction before it finally relaxes. So we can, if you move the cells further apart, they'll diffuse out more rapidly. If you put them closer together, they'll diffuse. Notice our motor end plate has those invaginations to increase the connection, so increase surface area. In these ways, we can change how long the neurotransmitter stays in that synaptic cleft, and the longer it's in the synaptic cleft, the longer it has a chance to have an effect. All right. Lastly, again, it's all about the receptors. If I have a cell that only has one acetylcholine receptor on it, and I dump a whole bucket of acetylcholine on this cell, is that going to have a huge effect?
Yeah.
Well, yes, with 1 million acetylcholines, definitely acetylcholine will be able to bind to that receptor, but it's only one receptor, one door. How much sodium can we get in one door? Probably not that much. Or let's go to the extreme. What if I remove that receptor? Whoops. What if I have a cell that has no acetylcholine receptors and I dump a whole bucket of acetylcholine on it? Am I going to have an effect then?
No.
No, exactly, right? If there's no keyhole, doesn't matter how many keys you have, you're not getting in, right? So all of these are things that can affect how strong the influence of the neurotransmitter is. And then we've already talked about how, like with curare, we can block it. With tetanus, we can activate it. We've done all of that stuff as well. All right. Like I said, last thing I want to talk about are the different types of neurotransmitters. Again, with over 100, we're obviously not going to identify all of them. But acetylcholine, as we mentioned, is the most common in the peripheral nervous system. And as we have now seen, it can be either excitatory or inhibitory, depending on the receptor. But key there is in the peripheral nervous system. That means it's not the most common in the central nervous system. In the central nervous system, the primary neurotransmitters are actually amino acid based. Glutamate is the most common, and so let's just do it this way. Most common in the central nervous system. Glutamate is the most common excitatory neurotransmitter in the central nervous system, and gamma aminobutyric acid, which, and again, as you always know, I don't like abbreviations, but this is one I will give you. GABA. You may use the term GABA. You don't have to memorize gamma aminobutyric acid. GABA is the most common inhibitory neurotransmitter in the central nervous system. There can be a larger, what we call biogenic amines, protein-based hormones. Some examples of those include epinephrine and norepinephrine. What's the other fancy word for epinephrine?
Is it adrenaline?
Yeah, adrenaline. Epinephrine and norepinephrine are the exact same thing as adrenaline and noradrenaline. Right? These are our stress hormones. These are the ones that become active when we have to fight that bear with an axe. Dopamine. Remember, we talked about dopamine when we talked about Parkinson's. Dopamine levels also seem to influence a lot of social anxiety-based disorders. So they're looking at dopamine as a very, very important central nervous system behavioral neurotransmitter. Here are two good ones: endorphins and substance P. Where do endorphins come from?
Exercise?
Yeah, exercise is one place you get it. Where else do you get them? There's the three big. Exercise, what else? Where's the other places you get endorphins? Come on, I know you guys know this. All right, I'll give you the second one. Exercise, love, and what's the third?
Is it the feeling of accomplishment when you finish something, sports, won?
Well, I think that got the exercise aspect of it. Absolutely, the exercise from that. Also, you get endorphins from being in love. And if you're not active and not in love,
Sleeping?
what else? Say again?
Sleeping?
Sleeping's not a bad guess. Nope. What's the other thing that can give you the same feelings as love? Really, none of you know this.
Happiness?
Not just any food, but what food in particular do you need to eat to give you endorphins?
Chocolate.
There we go. Chocolate. There you go. Love, chocolate, and exercise. Those are the things that will give you endorphins. Absolutely. Yes, Lori, you had a question.
Yes, going back to the biogenic ones, the epinephrine, I know is that what you use like if you have an allergic reaction?
Yep, absolutely.
Okay.
I have a pen filled with adrenaline that we lovingly call an EpiPen that we carry with us everywhere we go to with my daughter. Absolutely. Again, epinephrine is a stress hormone. So again, take the instance of why I carry it around. As I've said before in this class, my daughter has an extreme peanut and tree nut allergy. So she's allergic to nuts. And with nuts, there is the risk of anaphylactic shock. In anaphylactic shock, you're basically getting an inappropriate immune response. And there are two main severe concerns that you can have. One is it causes systemically, basically throughout the body, a dilation of blood vessels. And so blood pressure drops. And if blood pressure drops, it can be hard to get blood to the brain. The other thing that happens is this inappropriate immune response causes the airway to produce a large amount of mucus. And so that increased mucus production makes it hard for the individual to get air. So you can't get air, you can't get oxygen, you can't get blood to your brain. Very, very dangerous situation. So what do? You stab them with the EpiPen, right? The goal of giving them that adrenaline rush is to basically do two things. It causes a systemic constriction, constricting blood vessels, bringing blood pressure back up, and it causes the smooth muscle in the airways to dilate, hopefully allowing air to get in. Well, hyperventilation is a condition where you get a dangerously low amount of carbon dioxide in your blood. So that can be a problem that way. Yeah, it can be psychosomatic. There are other things that can cause that as well. So hyperventilation, so inappropriate breathing or too rapid breathing can cause your carbon dioxide levels to drop too low, affect the pH of your blood, also make you dizzy and pass out. All right. So again, yes, so those EpiPens basically mimic the stress response or stimulate a stress response. Yes, Dimitri, you had a question?
Yeah, not really a question, just a really quick story. So my teacher one time tried showing us, like she had an EpiPen, and she tried showing us, how you use one. And instead of using one that has already been used, she had a new one and she mixed the two up and in front of the whole class injected herself with an EpiPen and was hyper for the rest of the day. So yes,
yeah. They actually have testing, they practice ones that aren't actually used ones that are empty. So I mean, unfortunately those kind of things happen, right? Yeah, you mistake an empty EpiPen for a full EpiPen. You mistake a real gun for a taser gun. I mean, these accidents happen, right? That's just life. All right. Notice I mentioned endorphins here. We talked about that, but notice also substance P is another one. Now, many of you were able to figure out at least some of the things that endorphins come from. Substance P is the primary neurotransmitter in our pain system. That's what the P stands for with that. And then there's some more unexpected things that can be neurotransmitters. ATP can be a neurotransmitter. Gases like nitric oxide, and I mean nitric oxide, not nitrous oxide, right? Nitric oxide is what you get at the dentist office or at that rave on Friday night. Nitric oxide, though, has a similar effect where it causes a dilation of blood vessels. So it has that same type of effect that way. And again, your book's got a nice table that goes through many of these. All right.
I have a question.
Yes.
How does substance P relate to arachidonic acid?
I'm not familiar with arachidonic acid.
It's the chemical that like inhibiting arachidonic acid is what NSAIDs do.
Oh, I don't know the relationship of them off the hand. All I know is, like I said, substance P is one of the main neurotransmitters that the neurons use. I'm not sure how that would. My guess is that it's stimulating the nerves to release substance P so that you sense pain. So if you can block that, then that's going to lessen the pain sensation that you have. But I'm totally guessing.
Okay, but things like GABA, people take GABA and like gabapentin to combat that, correct?
Pain sensation?
Gabapentin is used for nerve pain commonly.
Yeah. Again, because it's an inhibitor. If we can stop the neurons from firing, we stop those pain pathways from carrying through. Yes, but that GABA is more in the central nervous system. Substance P is more in the peripheral. All right. Excellent. But yeah, similar pathways. And pain pathway is one of the least understood one. It's a very interesting one, but it's a very challenging one to study. Surprisingly, many human volunteers are not willing to submit themselves to multiple repeated pain so that we can figure out how their brains are communicating. All right. So like I said, you have a nice table in your textbook that talks about these and many, many more. And with that, we are done with the things that I wanted to talk about from a lecture standpoint today. So excellent. We are still, well, that's between you and the researchers. So you can take that up with them. That is what I wanted to cover today from a lecture standpoint. So let's take our first break and then we will come back and we will pick up where we left off. So let's go ahead and that.
Can I ask a question?
No questions. Yes, of course.
So we didn't talk about smooth muscle. What effect does acetylcholine has? Is it inhibiting or?
No. So acetylcholine, it depends on the location of the smooth muscle. But for instance, in the erector pili muscle, that is one of the locations where we use acetylcholine and it causes it to contract. Most smooth muscle is stimulated primarily by, well, actually, no, so yeah, I think in some locations it's going to be excitatory, in some locations it's going to be inhibitory. So the erector pili muscle, for instance, it is excitatory. In the stomach, it is excitatory. But in some of, for instance, the valves of the digestive system, it is inhibitory, allowing it to relax, increasing the flow of food.
It has nothing to do with like if we can voluntarily or involuntarily control the muscle.
Correct. No. Yeah. No. No. That is correct. Great question. No, it is not related to that. No, it is different organs. It's going to have different effects.
Okay. Thank you.
All right.
Sorry, last unit. We were going to talk about coffee and cigarettes, but this is kind of related. But is this the same reason why caffeine and nicotine cause, like, mass or like fast digestion, because it relaxes the your bowels, basically?
Yes. So yes, nicotine is what we call an acetylcholine. We'll talk about this at the end of the semester. We got a few lectures to get to this, but basically, so let's say it this way: it mimics the effect of acetylcholine. So the same way we talked about how tetanus will bind to a muscle cell, the cation channel, like the acetylcholine will, and hold it open and cause that rigid paralysis. Nicotine also can mimic the effect of acetylcholine. Now, it doesn't bind and not let go the same way tetanus does, but when you have that nicotine, often it causes your muscles to be a little bit more excited, to be a little bit more edgy or jumpy as a result of that. It does or alert, I guess, that way. It does increase blood flow to the nervous system. There are some pleasure sensations that are stimulated by it, which leads to the addiction of it. So things along those lines, yes. So in that case, it does mimic the effect of acetylcholine. Caffeine has a slightly different effect on a different receptor. But
if you bombard your body with it and then you don't have any of it, that could be really bad.
Well, yeah, what you're talking about is right?
Yeah, you could get receptor fatigue and then it doesn't work so good, right?
Well, that's a great question. That is one of the problems with drugs in general. You have a cell that has receptors, right? So you have these nicotine receptors on the surface of the cell. And so the very first time you have some nicotine, it's going to come and that nicotine is going to bind to these receptors and you're going to have some type of effect, right? But then, as we talked about, the expression of these receptors can be dynamic. So as you continue to take more and more nicotine, then what happens is you start to downregulate the number of receptors. And so what happens is now you need to get more nicotine in your body to have the same effect that you had before. And then you downregulate it even more. And now you have to have even more nicotine in your body to get the same effect that you had before. And that's true both for nicotine and for many other medicinal and recreational drugs, is we have this dynamic downregulation of the receptors. So the effect of it becomes less and less, and therefore it takes more and more to get the same high that you got the first time. And that's the problem with these kind of things. That's how you get people who, you know, smoke their very first cigarette and are puking and everything as a result of that. And then, you know, 12 months later, they're up to 2 packs a day. I'm pretty sure they do these kind of things with high schoolers about vaping and those types of dangers associated with that, at least more and more anyway. All right, excellent. Any other questions on this stuff?
Thank you. I didn't mean to hijack everything, but yeah,
No, I'm happy to
it's
answer
really
these questions.
And also like with vaping,
Yeah.
E-Valley, the e-vaping lung disease is actually like becoming more and more and more a big deal. People are, it's really bad.
Yep. Yeah. Well, and yes, education is an important way to do this, because you have the same problem that you have with cigarettes. You can have all of the information out there, but the addictive nature of it makes us, you know, the addictive nature of many things makes us do things that we don't want to do. And so, it takes a community, it takes education, it takes lots of things to help people to be successful with this. It's really easy to say stop, right? But it's harder to do those things. All righty. But again, that's a deep philosophical class question for another time. All right, let's go ahead and take our first break, as I mentioned. Oops, that's not what I wanted. We will come back from this, and like I said, we are going to switch gears and jump back into our brain anatomy handout. So if you don't have that printed out, if you don't have that handy, make sure you go and get that. It is 1:28 right now, so we will come back at 1:43.
Professor, what handout are you talking about right now?
Hold on. Although I guess it doesn't matter now, because as soon as I go away from this, it's going to disappear. So in your modules, under the nervous system, there is, under the lab handouts, the nervous system histology, but also the central nervous system anatomy. This is the handout that you are looking for. This is the one that, if you look, this is the stuff that we'd gone through. We'd gone through all of the general structures of the telencephalon, some of the specific structures of the telencephalon. And we left off last time. We had gone through the frontal lobe and had just started the parietal lobe in our discussion of the different specific regions or areas you are going to be responsible for. As I mentioned, this handout and the histology are going to likely be about 50% of your lab exams. So again, hopefully you are working on that. Well, no, that's actually for today's lecture. Those are some things, those things that were there are things that we're doing for today's lecture. So those are, yes, some YouTube videos that we will be using today as we discuss this material. So I'm not doing anything sneaky. That's what we're talking about. Excellent. All righty. So again, meet you back here in 15 minutes. So like I said, we will restart at 1:43. All right, see you in 15 minutes. So any questions before we dive in?
Not yet.
Excellent. All right, perfect. So we left off last time. We were working our way through our telencephalon, which, as we know, in the mature brain is the cerebrum. We know the cerebrum is divided into at least 4 and maybe a half of a lobe thrown in there as well. And they are based on the bones: frontal, parietal, occipital, and temporal. And we had left off last time, we completed all of the regions, all of the areas that you were responsible for in the frontal lobe. And we had jumped across the central sulcus to the postcentral gyrus, where we talked about the primary somatosensory cortex. And we saw the 2 homunculi that you would see there and everything else that we talked about that as well. Now, as I mentioned, these Brodmann numbers are useful because it helps us to identify the things that we were talking about. And there's a surprising symmetry between the consistency of structures and functions, but it's not perfect. If you notice, for instance, here, there's kind of this whole big region that involves 39, 40, and even a little of 22, basically right where the thumb reaches the hand. In fact, I've got this pretty picture here that does a nice job of showing this. And this region is an area that is known as Wernicke's area. And of course, why is it called Wernicke's area? There you go. Good old Bob Wernicke was the first one who identified it and described it. And again, it is commonly referred to as Wernicke's area. Remember, we talked about damage to these speech-related areas, and hint, this is a speech-related area. We refer to as aphasias. And damage to this area is indeed known as Wernicke's aphasia. But this area is also known as the general interpretive area. This is where we process and make sense of speech, both speech that we are presented with and speech that we are going to produce. Remember, we had already talked about Broca's area, which, as we can see, is over here. That is the one where we produce speech. And as we talked about, if someone has damage to that area, they may have difficulty producing meaningful cadence and meaningful speech. But they understand it. If you ask them to raise their left hand, yeah, I know it's my right hand, but you get the idea. They're able to follow directions. People who have problems with Wernicke's area, this general interpretive area, they have difficulty both understanding and producing meaningful speech. What's interesting is their speech can still have the same expected cadence to it, but it won't always necessarily be meaningful words. They may add extra syllables or use words improperly. People who have Wernicke's aphasia, it is often referred to as word salad that they do. And I actually have this great example that I've used for many, many years. It was one of the tabs that you guys saw open. So let's take a look at that here and watch this video.
Boom.
What's this?
The big anchor right there. And
Can you guys hear that?
Yeah, yes, I can.
Okay.
the football.
And the football?
It influenced the football right here. The cause that it comes in, see?
Yeah.
The end of it, see? Yeah. Oh, we have all of these, see?
I don't understand.
Well, it's quite pliable in the other, see? You don't understand it, but it flies in the long tree comes from there, and it comes source from the country out here, see? All the under it, see, it comes up, and finally come out here and goes out, see? Get it?
Sort of.
Yeah, it comes out here, see, it comes out here, here, and here, but it comes in, and it comes out, and bing. That's a good flow, see?
That's a good what?
That's a good flow. Flow goes fly, see, it flows cold, or it gives, or kicks out, or bing, or whatever it is. We has no football about it out. It's just pulling, see? But they feel it. Oh, I guess you meant you have beds. See, I don't know.
Where do you live?
Where do you live? In your live? You're up here, see?
Where do you live, Doc?
Where do you live?
Where do you live?
Where do you live is, well, if there's a run here, around here. You?
You used to be a dentist.
Yes,
Where?
right here. the rest, the rest of the here, yes.
And how many years were you a dentist?
How many? Holy Christmas. Oh, about 100 and, let's see, 130, about 40, no. Gosh, what number is it? One plus 5. Wait a minute, no. It's a pretty close to get to this one day. It's about every year off, see. It's claws. Oh, it goes for about over 30, see, or 130 for down, see, in front of out for if they're out for it goes against her, and then it comes back to the against her 4, and then goes out for the rest of the time. The third is not the servant, but the soldiers, where they have the soldiers of the same birds and the suburbs and all the first third river and watch out in the short side, and they cross through the sugar 2 hours, 2 high or low. Wow, God, these are terrible. Wow, these are terrible words.
These are terrible words.
They sure are.
Hard?
Yes.
What you're saying, you mean?
Yeah, that's what they do. They actually do them a lot harder. It's when they harder in hard go. And then when they go out, finally they quit, see? You have to have a time, don't you?
All righty, so let's talk about that. So notice if you were at a Starbucks sitting at a table and that conversation was going on behind you and you weren't really paying attention, you wouldn't really think anything about it. Because if you're just listening to it in the background, his words have inflection, it has the normal cadence of speech. It isn't until you start paying attention to what he's saying that you realize that what he's saying isn't necessarily making sense. Now, I saw some of your guys' comments, and yes, obviously this can be very challenging. If you notice, he shows a little frustration there at the end, talking about how horrible these words are, how they're here, and then when they come out, they just die. But at the same time, he makes a lighthearted joke about being a dentist, saying that he's a dentist in this and laughs at it for himself. This is a challenging condition because obviously he's clearly aware of that he's having some difficulty. Like I said, he shows that frustration at the end about not being able to get the words out. But no, this is damage to Wernicke's area. This is strictly caused by damage to Wernicke's area. At the same time, it's hard sometimes to fully appreciate how much they're able to understand and not understand. You can see that obviously he's having trouble saying words, but he also has trouble understanding them as well. And I think I've seen this many times. So again, these are subtle things you may not have caught when we worked through it the first time. But notice one of the questions she asks him is, where do you live? Right? Now, if I were to come up to you in the classroom and ask you, where do you live? Right? You may think about whether or not you want to tell me the answer to that question. And so while you're thinking about it, typically what you'll do is you'll turn the question around. Where do I live? Right? I say, where do you live? And you say, where do I live? But notice she asks him, where do you live? And he repeats the question verbatim. Where do you live? Where do you live? He says the exact same words she does because he's trying to make sense with them. Those words aren't making sense to him. He doesn't know what they mean, right? If you know what those mean, you can turn them around. Where do you live? Where do I live? Right? She says, where do you live? He says, where do you live? Because he's trying to make sense of those words. So again, it can be challenging to fully understand what they do and do not understand. Also, depending on the severity of the damage, for some of these people, they are able to write normally, but for some people, it also affects their ability to read and write as well. The last thing I want to say about this is that this is another example of lateralization. This is a condition that in essentially 100% of right-handed people, over 90%, let's say it that way, have this Wernicke's area over here on the left side of the head only. And even left-handed people, over 75% of left-handed people on the left only. So then the question goes, what's over here on the right? It turns out on the right is spatial awareness. I think I saw someone write that they talked to a guy on the light rail that when they were in high school that had this kind of a problem, this word salad. It could have been something along those lines. I had a student, gosh, 5-6 years ago now, that her boyfriend at the time had been in a car accident, and he had a traumatic brain area to what would be Wernicke's area on the right side, that part right at the base of the thumb of the temporal lobe in the parietal lobe. And as a result of this, they lose spatial awareness for the contralateral side. So damage causes a loss of awareness of the contralateral side. If you asked him to draw you gave him a flower and asked him to draw it, his drawing would look like this. There's the pot, there's the stem, there's the heart of the flower, and there are the petals. Notice he wouldn't draw any of the left side of that flower. He doesn't perceive it. He often will not dress the left side of his body. When he's in the shower, he will not wash the left side of his body. He will often get frustrated of whoever it is that's standing too damn close to him on the left side. Hey, buddy, where are you? Get away from me. Because there's this arm and there's this hand and this leg on his left side. It's not that he's blind to it. It's he doesn't recognize it. He's not able to make the reference to whatever that spatial stuff is on that side. He's just not aware of it. And again, it's not 100%, but because it wasn't a complete damage to it, but these are the types of difficulties that he has as a result of that. So again, it's one of those things, this localization and lateralization, a specific region that has a specific function, and on the left and the right, they have different functions to them. All right. Questions on that?
Wait, what you just described was of the Wernicke's area, right?
Yes, Wernicke's area, absolutely.
Wernicke's.
That general interpretive
Okay.
area. And we talked about the aphasia and the lateralization for that spatial awareness on the right side. Okay. Now, that brings us to our occipital lobe. Our occipital lobe, remember, is basically this part back here. And our occipital lobe, in fact, most of the brain is actually involved in processing visual information. Yes, I will stand here and tell you with 100% honesty that by trade, I am a visual neuroanatomist. I am 100% biased. I love the visual system. The eyeball, I think, is by far the beautifulest organ in the absolute body, but you have over 1 million neurons carrying axons out of each eyeball, carrying information to your brain. Over 40% of the information you receive from the world around you is visual information. Over 60% of your nervous tissue is involved with processing visual information. We are visual organisms, and it is the primary way we process information. And not surprisingly, then, we have a tremendous amount of nervous tissue involved in processing and making sense of that information. And in our brain, it all starts here in this area 17. The very, very back of your brain, area 17, is where we perceive information from the visual information from the contralateral side of the body. Notice I didn't say from the left eye or the right eye. I mean, over here on the right side of the brain is everything over here in the left side of the world around you. This seems a little tricky how this might work. But we have this pretty picture that kind of shows this. Notice if you close one eye, you see stuff to the right of you and to the left of you. So both eyes receive information both from the right side of the world and the left side of the world. So what happens is some of that information, notice this information here from the left side of the world, from the left eye, has to cross to go to the right side of the brain. Whereas this stuff here on the right side in the right eye stays on this side. Whereas blue stuff from the right side, some of it crosses to get to the right. The stuff from the left stays on the left. So we have this partial crossing of axons that occur from the eyeball, allowing everything from the right to go to the right, everything from the left to go, everything from the right to go to the left, and everything from the left to go to the right. So it is the contralateral side of the visual world that we see with that, all right? So this primary visual cortex then is where we just that simply perceive light. Yes, Arthur, go ahead.
Yeah, I heard somewhere where like there's these goggles where turn your world upside down, and like if you wear that for like 3 days or whatever, your brain will flip that and you'll be able to see like straight or whatever. I don't know how to describe it, but
so no, I know exactly what you were talking about. So if we have time when we get to the end of this and we're talking about the sensory systems, one of the things that we'll learn is back in ancient times, there were these things called cameras, and cameras had this stuff called film inside of it. This stuff was light sensitive, and as light came in through the lens, that light was focused on the film, which was photosensitive, and it would basically imprint it with an image. One of the interesting things about the way that the lenses work is they take information, light from the top, and put it on the bottom of our retina. Things from the bottom go to the top of the retina. Things from the left go to the right. Things from the right go to the left. So basically, our retina of our eye receives information that is upside down and backwards. So you're absolutely correct. There were some scientists who were interested about this, saying that it's really weird that the brain sees the world upside down, but it fixes it for us. The information coming in from our retina is upside down and backwards, but our brain fixes it. So what if we make these students wear these lenses 24 hours a day, and that turns the world upside down and backwards onto their retina, so everything is normal looking on their retina, and then what is our brain going to see? And lo and behold, not surprisingly, they saw the world upside down and backwards. They got motion sick, they fell down walking, they had trouble eating and drinking, and they definitely couldn't drive a car. But then you're right, after a few days, something truly remarkable happened. Their perception of vision actually switched. Somehow their brain figured it out and was able to reverse the image. And then they saw the world clearly and normally again. Awesome. They wore the goggles for another couple days or so, actually about a little over a week or so, continuing to do that. And they saw everything normally and everything was fine. And then the day came for them to take the goggles off. And when they took the goggles off, their vision was suddenly upside down and backwards. So now without the goggles, the world was screwed up. And you had some incredibly panicked graduate students trying to figure out, oh my God, what the hell are we going to do? We broke these students. Luckily, after about a day, the vision went back to normal again. And it was really awesome. Obviously, it shows that the brain has the ability to perceive and comprehend and modify what we see, but we still don't know the mechanism by which that occurred. We don't know where in the brain that information got switched so that we made sense. It's one of the awesome things about the nervous system is it's still the Wild West. There's still so much to learn. Yeah, that's what college was. In the 70s, when you went to, when you graduated high school, you did two things. Oh, well, you did three things. You went to some trade school, you went into the Army, or you went to a 4-year college. And at that 4-year college, your first year there, you were required to take a Psychology 1 class. And that Psychology 1 class was basically just an excuse to torture students. They made them wear goggles that saw things upside down. That whole Stanford prisoner's dilemma thing that happened there. They were able to do all these, sociological and physiological manipulations to see what would happen. Right? Basically, they were torturing freshmen in the name of science. All right. Primary visual cortex is where we perceive light, period. Again, one of the easy ways to understand this is if you damage this area, you're blind. You no longer perceive light. Now, hopefully that hasn't happened to any of you, but maybe some of you have been knocked in the head or been in an accident. And as you've been in that accident and your head whipped around, maybe your brain smashed up against the skull in the back. When you talk to people who've had that kind of experience, they often talk about seeing stars. That seeing stars is because when that occipital lobe and that primary visual cortex pushed against the skull, it stimulated those areas and you saw stars as a result of it. So this is just where we perceive light. But yeah, exactly. It's not uncommon for this to happen to people. Yeah, well, you saw stars because you smashed your visual cortex against your skull. But we need to make sense of that information. And where we make sense of that information is what we call the association cortex. So typically primary cortex is where we get the raw data. Like light. Association cortex is where we process the data. We see lots of examples of this. Probably one of the easiest ones is this area right next to primary visual cortex, area 18. Area 18 is part of our association cortex. And that is actually where we perceive color. So for instance, if you have damage to just this area, you would have difficulty perceiving color. And it gets more complicated than this. Visual processing cortex goes way up here towards the parietal lobe, comes way down here towards the temporal lobe. Like I said, over 60% of the nervous tissue is involved in processing visual information. And what's interesting about this is that there's primarily two main pathways of information. The first is what is called the WARE pathway. The WARE pathway is where we perceive motion, perceive where something is located in space. Now, this is the name of an area I'm not going to hold you responsible for, but there is an area up here called MST. And people who have damage to MST don't perceive optic flow. Now, what do I mean by optic flow? Well, if you were to sit in your chair like I was and to spin yourself around, what do you see in the world as you do that?
Everything around you.
Yeah, the world slides by you. Or when you move forward in space or back in space, when you move forward, basically the world expands around you. When you move backwards, the world moves forward. That is what we call optic flow. And people who have problems with MST don't perceive optic flow. So you ask, well, what would that possibly feel like? Well, when we were talking about being at that rave on Friday night, maybe you happen to be in a room with a friend where they've got a strobe light going off. And when your friend across the way is waving to you across the room while that strobe light is going off, is that what it looks like they're doing?
No.
No. You see their hand here, and then suddenly their hand's here, and suddenly their hand's here, and then suddenly their hand's here, and suddenly their hand's there, right? You don't see movement. It's just a bunch of basically stop actions, stills. That's what people who have damage to MST feel like. Now, can you drive if you're constantly living in a strobe light?
Really?
Probably
No.
not a good idea.
Yeah, absolutely not. Absolutely not. Heck, can you pour a cup of coffee if you have damage to your MST area?
No.
No, because the coffee's here, and suddenly it's overflowing. But as we know, humans are very ingenious. And in fact, people with damage to MST do pour their own coffee. How do they do it?
Yeah, I have time in between frames to think about their
I
trajectory
guess
and
that's
speed.
one way. If you timed it, if you know it takes it 3 seconds to fill it, but you got to be really careful about how you're tilting it. What's an even
Do
easier
people-
way than that?
Is the sound of how full the cup is?
Sound would be one way to do it. There's an even easier way than sound, though.
The weight of it?
You put your thumb in the mug. If you put your thumb over the cup of the mug, you just fill it up till you feel the coffee come up to your thumb, and you know you're done. Right? We're clever individuals.
That sounds painful.
All right. So MST may be, well, again, I'm not saying you immerse it in it, but when it comes up and you feel it, that's fine. This ability to perceive motion, like I said, is an important function of our nervous tissue. Another example that we see of this, let's go ahead and stop. Hold on, let me do that because I think that'll help. Another example of where you see this is in an illusion that is known as the waterfall effect. Our perception of motion is the perception of movement, right? I don't know what a GPU is. Perceive there. So what we're going to do here is there's going to be a video. You are going to stare at the eye in the center of the video. Oh, yeah. Stare at the center of the video. Try not to blink. Try not to look away for about a minute. And then when the video ends, you're going to either look at your hand or look at the room around you. All right. So again, try not to blink. Try not to move your hand, move your eyes. Try to watch this for a minute. So here we go. We don't need the sound. Here we go. So try to watch it in the center of the screen. Try not to move your eyes. Almost there. Now look around the room and what do you notice? What do you see?
I see an eye,
Is that all you see?
like the letter I, and everything's spinning.
Yeah, everything's spinning. Absolutely. What you're seeing that spinning is because of that after effect, what we call the waterfall effect. right? You may never have looked at one of these hypnotic images before, but maybe you've gone up to Yosemite and you've stared at a waterfall for a while. And as you've watched that waterfall, one of the things that you may notice is the land next to the waterfall looks like it's starting to move up. And that's because what's happening there is you are fatiguing the cells that are noting motion. So for instance, you're fatiguing the cells of the waterfall that are moving down. So when you look at something that's moving still, all your cells fire except for the down ones, which are tired. And so as a result of that, they go up. Maybe you're looking at it too close or something like that. You can try this on the own. The other cool thing about this, and this is something that you can find on your own, it's called a motion after effect. So try this motion after effect. But the other thing that is cool about this is this happens in the brain. So what happens is you can stare at that image with just your left eye and then look at your hand with the right eye, and it will morph as a result of that because the change that is occurring there in the brain. One last example. Down here in the what pathway is where we make sense of visual information that we see. There is an area known as IT for intertemporal, but that intertemporal area is where we perceive complex objects to understand what it is. There's a very famous individual who had damage to this area and has been experimented on for many, many years. And one of the classic examples of this is he was shown a glove on a stand. So there was a glove on a stand. It was striped in yellow and blue. And he was able to describe what he saw, the colors of it, right? The shape of it, things along those lines. He was able to do that. Clearly, he could see it and perceive it. But when they asked him what it was, he wasn't able to identify the object. So he stared at it for a while. And as he stared at it for a while, he saw these extensions on the bottom and thought, oh, this must be a change purse. You know, one of these is for nickels, one of these for quarters, one of these are dimes, one of these are for pennies. Then they handed it to him. And when they handed it to him, he felt it. And then when he had that tactile sensation, he was able to realize it was a glove. He's married. But if his wife is just standing there, not moving and not talking, he can't recognize her. The second she speaks, he can recognize her. And sometimes when she walks, he can recognize her by her gait. But if she's just standing there and if she's not talking, he does not recognize her. Many books have been written about him. One of the most famous was The Man Who Mistook His Wife for a Hat. Right, and it's about this person who has this IT damage as a result of it. So as you can see, some of this accessory processing tissue can be incredibly, incredibly specific on the information. But the take-home message is, again, primary is where we get the raw data. Association cortex is where we make sense of it. And notice here in the temporal lobe, we have the exact same thing. In the temporal lobe, basically the central sulcus points almost right to it, is our primary auditory cortex. This is where we get the raw data on sound. And it is surrounded by 42 and a little bit of 22 here. It is surrounded by association cortex. And that association cortex is where we make sense of that information. You realize that it is an oscillating sound and it is increasing in amplitude. And so you realize, oh, that sound is a siren and it is coming towards me. I should get out of the middle of the street. So whether it's vision, whether it's hearing, we have primary cortex where we get the raw data and the association cortex where we make sense of it. All right. Questions on that?
Yeah, wait, what was the raw data in the auditory?
primary auditory cortex is where we get the raw data. And that's basically right here. Like I said, almost the central sulcus almost points right to it. So it points to the beginning of it. And then, like I said, the association cortex basically surrounds it like that.
I have a question.
Yes.
So I know the average size of the brain, like similar, there is not how do I say this? Like there is no people with big brain or small, but I've seen a lot of people that had like flat forehead, and some of them have round one. Does that mean their prefrontal cortex is larger and functions and better, and it affects their personality or anything?
That is a great question. And in fact, there is a field of study, I say study in quotes, of pseudoscience called phrenology, where exactly what you're talking about is one of the things that we thought occurred. Because we figured, after all, when you use a muscle, that muscle gets bigger. So if a person is kind, the part of their brain involved with kindness gets bigger. When the person is more intellectual, the intellectual size gets bigger. And so they thought that by feeling the bumps or looking at the curvatures of the skull, we could get a sense of how whether a person is kind or a liar or, happy or sad based on the shape of their skull. And no, it turns out there is no correlation between the shape of someone's skull and the functioning of their brain. No. It's a great thought and a great idea, but it's not accurate. It's not true.
I have a question too.
Yeah.
There are people who commit these random weird crimes that are super inconvenient for them, like murder their own child or something. And there's the dilemma of, is it a mental disability? Is it like a chemistry in their brains messing up? Or is it an actual evil intent? Is that something people can see visually on a brain?
No.
Or not?
Yes, the evil brains have a curly mustache on them. So,
no, I meant is that like a deficiency or a malfunction that we can see, or is that something that's left to the soft sciences to figure out if someone's guilty or not?
So there is no such thing as, a particular wiring of the brain that if you have this particular wiring of the brain, you are going to be evil, right? Or you're going to make evil decisions or be a murderer or anything along those lines. That doesn't mean that there aren't meaningful morphological differences between brain anatomies, but the cognitive function and there's the answer to your question is so much more complex, because basically our behaviors are a combination at the simplest core of the genetics of how we're hardwired, but also the experiences we've had in our life and how we respond to those. And so there's so much more complexity to that. Yes, there are people who some biological disorders like bipolar can be caused by imbalances of neurotransmitters or deficiencies in neurotransmitters. Epilepsy is a condition with inappropriate neural firing. So absolutely, there are some biological causes for some cognitive deficits, but a lot of behavior can't just simply be explained by the wiring of their brain. So yeah, I mean, so obviously someone like that, your neighbor, obviously had some kind of chemical imbalance or some other type of problem. But again, that while there can be biological causes for those types of behaviors, again, it's not always necessarily, the sole cause. Again, you can't diagnose people's behaviors solely by the anatomy of their brain, and certainly not by looking at it. Maybe by looking at the cognitive patterns, how their neurons fire and stuff like that in a functional sense, but not just by looking at it. No. All right. So we've done our 4 main lobes, but remember, as we talked about, nestled here inside of the lateral sulcus is this big chunk of cortex known as the insula. I think it's fun looking at it in that previous view, but here we really get a sense of what it is and where it's located. It is this big chunk of gray matter inside of the lateral sulcus. And I've written down my definition of what it is here. Actually, we'll have the next one in a minute. This particular cortex, the insula, basically is the cortex, the gray matter, that is responsible for kind of regulating the activity of all of these nuclei. All of these nuclei and more that are found inside the gray matter of our cerebrum collectively are what are known as the limbic system. And I've written this definition down because I want to read it to you precisely. The limbic system, which is basically located here deep in the cerebrum, is a functional group of nuclei, a bunch of nuclei that work together. All right. They function for emotions, memory, memory storage, and memory retrieval. And here's the part that I wrote down because I wanted to make sure that I got this. It links conscious thought and unconscious function. What do I mean by that? I did say those words out loud, right?
Yeah.
Is it to, like, you have a thought, it connects your thought to performing that thought?
Can be performing that thought, but it doesn't necessarily have to be. Think of it this way. When you come stumbling home at 3 o'clock in the morning on a Wednesday, completely 3 sheets to the wind, fumbling with your keys at the door to try to get in, making a tremendous amount of noise, waking up the baby, waking up the dog, waking up the neighbor. And when your loved one opens the door, do they even have to say a word? Or is it merely not just the look on the face, but that blood vessel that is pulsating on their forehead that tells you how much they love you and how happy they are to see you? Right? Do the conscious thoughts we have, can they affect our body physiologically? If you are thinking happy thoughts or sad thoughts or angry thoughts or horny thoughts or any of those types of things, does that affect the function of your body?
Yeah.
Yeah.
Like your heart and stuff like that.
Absolutely. It's the whole point of a lie detector test, right? I connect a bunch of sensors on you that measure your blood pressure, measure your heart rate, measure the conductivity of your skin, which tells us how much sweat you're producing. And I ask you what your favorite color is. And I ask you, do you like puppies? And then I ask you, where were you on the night of the 3rd? Right? Because I want to know how you're going to respond to those. And if you're scared or if you're feeling guilty, does that change your heart rate or your breathing rate or any of those types of things?
Yeah.
Yeah. And you don't have to be a policeman, right? You can have a far more noble job like a professional gambler, right? What are the things that professional gamblers are looking for in you to help them when they're playing against you?
Face.
Yeah, they're looking for
Or
what
Body language.
Yeah, tells. What they're looking for tells, right? Your body language. Are you nervously, moving your feet or clicking your fingers? Are you moving around in your chair, right? They even look at your neck to see if your carotid artery is pulsating. If your carotid artery is pulsating, that could be an example of you being excited that you got a really, really good hand, or maybe you're lying and making a really, really big bluff. Right? That's what we're talking about here. The ability of what we think about to actually have a physiological effect on our body. That is what our limbic system does. And our insula plays a huge role in this. In particular, the insula, this bit of cortex here, plays an important role in, this is also where pain sensors are located, anger, This is the cortex involving our sense of smell. If you think about it, our sense of smell is the one that is most closely related to our emotions. And the other one, and this kind of goes to things we were talking about before. Yes, we'll talk about that in a second. This is the site of addiction. Right? As we were talking about before, we could all decide after class today that we are all going to smoke a cigarette. All right, so we all smoke one cigarette after class today. By the end of the week, are every single one of us going to be up to half a pack a day?
Yeah.
No. Some people can smoke one cigarette and it doesn't bother them at all. Some of them, they smoke one cigarette and they desperately want another. Or maybe instead of cigarettes, it's a drink. Or maybe instead of a drink, it's gambling. They actually have looked at professional gamblers, where basically what they'll do is they take people, not professional gamblers, but people who are addicted to gambling, and they put them underneath one of those magnetic resonance imagings where they look at changes in blood flow to the brain. And so what'll happen is they put a normal person in there, and then they put someone who's addicted to gambling in there. And they have them play a game of chance, where basically they say heads or tails on a coin and flip the coin to see whether they're right or wrong. And what they find is that people who have a gambling addiction get a much larger activation to their insula to somebody who doesn't have a gambling problem. So addiction is absolutely something that can be biological in nature. The activities that someone is addicted to can cause activation of this area, which is important because, yeah, if we can find ways of blocking the activation of this, they will lose that enamor associated with it. And so it is a potential biological way of combating addictions. To get back to the question,
happen?
I'm sorry?
Oh, sorry. Would that happen only if someone actively had a gambling addiction, or could that happen if someone has an addictive personality but no active
I don't know if they looked at those types of generic individuals that quote unquote have an addictive type of personality. But my guess is that people who have that probably have more things or have a more active insula. So that something, as long as something is stimulating that area, then they're getting what they need. To get back to your original question about a lie detector test, the reason the lie detector test works is because you're scared or embarrassed or worried about what you're thinking about. Someone could be lying about what they believe without being aware of it, right? If you don't know you're lying, if you thought you saw that they showed you the 5 of clubs, but they really showed you the 7 of spades, but you thought it was the 5 of clubs, then you can sit there on a lie detector and say, yes, they showed me the 5 of clubs. I'm 100% certain about it, because in your mind you are, right? Many of the original lie detector tests just looked at things like heart rate and blood pressure. So one of the things that people would do is they would put a thumbtack inside of their shoe, so that when they asked them a basic, simple question, they would push down on that thumbtack so that they would be in pain, which would accelerate their heart rate and their blood pressure. And then when they were asked the serious questions, they would remove their toe from the pain, and so that would calm them down a little bit from there, so they could mask it that way. Most of the newer lie detector tests now also measure movement of the body. So if you're constantly moving your foot every time they ask you a question, they're going to catch that. So it's gotten more professional, more sophisticated. But as someone also said, if you are a sociopath, if you aren't nervous or concerned about the fact that you killed somebody, then you can easily lie without having that biological response as a result of it. Yeah. Well, it's one of the reasons why they talk about how eyewitness testimony is one of the least accurate of all testimonies, because often what we see is very much perceived by our own worldviews. So yes, that's something as well. Absolutely. But even if you've been tricked by your worldviews and you're not aware of that, then yeah, you easily pass the lie detector test and say, sure, I saw Billy kill her, even when it wasn't him. All right, let's talk about some of these nuclei located here in our limbic system. Talking first about this almond-shaped one, which is where it gets its name right here, the amygdala. Notice the amygdala is very deep and medial in our temporal lobe. The amygdala plays an important role in our association with memory and emotion. In particular, having to do with anger and fear. Right? One of the examples, a great example of this, for instance, is that monkeys. Monkeys, rhesus macaque monkeys have a very social hierarchy to their clans, or whatever you call a group of monkeys. And they primarily communicate both vocally, but also visually. Basically, monkeys have two main types of facial expressions. They have a very aggressive facial expression, and they have a very submissive facial expression. And so, not surprisingly, monkeys are very, very good at detecting these. We know this because they'll take a monkey and they'll sit him in a chair, and they'll give him two buttons to push, and they'll show them all these pictures of monkey faces. And if they show an angry monkey face, they push button 1. If they show a submissive monkey face, they push button 2. Typically, while they're doing these activities, every time they push a button, whether it's right or wrong, they get a little squirt of juice. Monkeys love juice, so they'll sit there for hours and do this activity. And they are really, really good at it. Unless their amygdala is damaged. If their amygdala is damaged, then they have difficulty recognizing fear and anger. Now, hopefully, you've never been strapped to a chair and shown monkey faces, but the other common name, or at least the one we use for it in our house, is the amygdala is also what we refer to as the worry monster. It's what helps us to recognize fear and anger. Right? So for many little kids, they can have a very vigorous worry monster where they're afraid that the boogeyman is going to break down the door and steal their toys or kill their dog or steal their mommy and daddy. And that's a good thing, right? When we were out living in the fields, sleeping in trees, bears with axes roam the countryside. So when you heard a branch snap, it could very easily be a bear with an axe coming to eat you, right? But the creaking of the floorboard now is probably just grandma going to the bathroom, not the boogeyman coming to get you. But that's not what your amygdala is going to tell you. Your amygdala is going to tell you it's the big bad monster coming to eat you, right? So what do? You have to get out of your worry monster. You got to get into your frontal lobe, right? Do math. Come up with an animal for every letter of the alphabet, right? Do these types of tricks to get out of your worry monster into your cognitive frontal lobe so that the worry monster can let you, so it doesn't keep you from sleeping at night. All right? Questions on that? I wouldn't say that. I think that my guess is probably more it's from a psychological standpoint. When we're scared and once we know we're okay, that sense of fear makes us feel very vulnerable. And most people don't like feeling vulnerable. And so once you no longer feel vulnerable, you're angry about the fact that you were made to feel vulnerable. So my guess is it has more to do than that, than just the amygdala itself. All right. None of the books have the best picture of this, but I do like this picture from your textbook. Basically, what we have here is a partial sagittal section. We have a partial sagittal section through the brain, but they've left the temporal lobe intact. And when they've done that and peeled away, what we see is that there's this big red globular thing. Now, it looks like it has this big knuckle at the end, but what actually happens when we actually look at the hippocampus, it has this big belly, and then it comes to a kind of curled structure, which is actually where it gets its name. Hippocampus means seahorse. So if you think, look, it looks like the tail of a seahorse, and that's where it gets its name. This hippocampus is the location in the brain where you convert short-term memory into long-term memory. What do I mean by that?
Like retention?
Yeah, kind of part of that is retention, right? If I told you to remember three things, a cat, a boat, and a car, what are the three things I just said?
Cat, boat, car.
Cat.
A boat, and a
Excellent.
car.
Now, if I gave you a sheet of multiple choice questions, simple math, 2 plus 5, what's 2 plus 5?
7,
What's 7 plus 3?
10,
What's 1 plus 1?
2,
What are the three things that I asked you? Do you remember?
7, 10, 2.
cat, dog, car.
Yeah, cat, dog, car, right? Excellent. So you're able to do that. You're able to both keep information in your working memory, which is your short term, but you're also able to put it into long-term memory where it can be retrieved at a later date, right? And that's what this conversion is. There are individuals who have damage to their hippocampus who aren't able to do this. One of the most famous of all is a gentleman who's only known as HM. He was camping, and while he was camping, it started to rain. So he packed up his camping gear, put it into the car, and then slept in the car. And the problem is one of the things he brought with him was a charcoal barbecue. And he ended up getting carbon monoxide poisoning and got damage to his hippocampus on both sides. I think in like 2004, they had like a 25th or 30th anniversary of working with this guy. Actually, one of the instructors at UC Davis was one of the first ones to work with him and worked with him for over 30 years. And every single day when he walked into that room, he had to introduce himself to him again. If he left for lunch and came back in the afternoon, he would have to introduce himself to him again. This individual, HM, has not been able to make any long-term memories since the accident. He has working memory. If you tell him to, remember ball, cat, dog, right? He can remember ball, cat, dog. But then you give him, yeah, there you go, exactly. Then you give him, not quite them, but more the what's her face character, Drew Barrymore character. You give him those math problems and he does a sheet of math problems and you ask him those three objects and he can't remember what they are. There you go. Exactly. 50 First Dates is a classic movie of an individual. There's another equally good, but completely different movie involving someone who has damage to their hippocampus. Yes, 50 First Dates is a comedy involving that. There is a great drama involving that. Does anybody remember that movie?
Is it the one with Channing Tatum and some other girl where she like gets in a car accident and forgets everything before?
Oh, is that I'm not aware of that one. I was thinking of an older one. I was thinking of a Guy Pearce movie from back in the day. Got to remember I'm old. Memento? Anybody remember the movie Memento? A police officer, a bad guy, breaks into his house, kills his wife. He has severe bleeding. He's injured and ends up getting damage to his hippocampus. Cannot remember anything that has happened since then, but is not able to make new memories, but wants to solve the mystery of the murder of his wife. And so he takes pictures and writes notes and gets tattoos on himself and does all these things. The movie is shown asynchronously, kind of the way he sees the world. It is an amazing movie if you've never seen it, and I strongly encourage you to check it out. But it's another example, great example of a movie of someone who has this hippocampus.
What was it called again?
Memento.
I forgot. I actually watched that in a philosophy class.
Yeah, it's an amazing movie. A really cool movie. A really good perspective of this type of condition. All right. Again, we've learned a tremendous amount from people like HM. Not surprisingly, HM has a tremendous amount of time on his hand. So one of the things that he does is play a lot of video games. So after lunch, you walk in. Of course, first thing you have to do is introduce yourself to him again. And then you'll show him a video game cartridge. Have you ever played this game before? Nope, never played this game before in my life. You'll put the cartridge in, you'll hand him the remote control, and he will proceed to get the high score on the game, swearing he's never played the game before in his life. All right, so obviously when people talk about things like muscle memory or functional memory, when they talk about these types of things, it's because of accidents like individuals like HM that have allowed us to gain some understanding and knowledge about the world around us and our brain and how it works. All right. Excellent. While we are here also, the other thing that we can see really coolly on both of these pictures is actually really all three of them. Notice underneath the cerebrum is this little stalk-like structure known as the olfactory tract. The olfactory tract, if you recall, ends at the olfactory bulb. Do I have my picture here? I don't. Let's do that. I want, oops, wrong. Excellent. So we have our olfactory bulb and our olfactory tract. And of course, what are our olfactory bulb and olfactory tract associated with? I mean, you can have, most memory issues aren't necessarily involved with the hippocampus. It has more to do with the organization of information. It's like having a file cabinet. If you just have one drawer in one file cabinet filled with papers, it's pretty easy for you to find them. If you have a piece of information, if you have 300 file cabinets, each of them full to the brim, and they're not in alphabetical order, there's no rhyme or reason to how you put the things in there, it can be very hard to recall information that way. So yes, all brain areas can deteriorate some with age, but most memory issues are more related to other higher order types things, or being able to recall the information. It is indeed a cranial nerve, but the olfactory bulb and olfactory tract are associated with the cranial nerve, the olfactory nerve. But what do you think they do?
Smell.
Yeah, they give us our sense of smell. If you remember, we talked about this way back when we were learning the bones. Remember when we were talking about our cribriform plate, we talked about how the olfactory bulb sits on the cribriform plate and the olfactory nerves come off of it into our nasal cavity, giving us our sense of smell. I think I told the story about my sister's boyfriend who fell down the stairs, ripped off his olfactory bulb, can't smell anymore. Right? That all sound vaguely familiar? I think I've told that story here.
Yeah, you have.
Okay, excellent. Yeah, so we've talked about this before. Well, now notice, as we also mentioned, our sense of smell carries that information directly to our lymphatic system. Our lymphatic system is our emotions, our memory. And so not surprisingly, our sense of smell is the one that is most closely associated with memory, right? When you've got that boy that's gotten away from you, every once in a while, it's nice to stare at his picture. Maybe you call his answering machine so you can hear his voice. But when you're really sad and lonely, you pull out that shirt of his that you've got in that Ziploc bag so you can open it just a little bit and get that smell of him and then close it back up. And it brings all the emotions and all the feelings back just from smelling his shirt, right? So that sense of smell, or maybe it's the smell of baked bread or an apple pie that reminds you of that summer you spent at grandma's house, swinging on the tire swing, you know, swimming in the pond or whatever it is. Our sense of smell, yes, I do. Our sense of smell is the one most closely associated with emotions. So again, our memories and our emotions, smells are the ones that trigger that the most. Yes, Arthur, you got a question?
Yeah, I've heard Disney does this with like their popcorn smell. They would purposely like like burn, like I don't know what they would do, but
No, you're absolutely
they
right.
have
And
the
it's not just popcorn smell. Yes, when you go to the different areas at Disneyland, different areas have different scents that they are sending in to enhance the experience. Absolutely. Right.
Yeah, and they purposely did it like to mess with us psychologically too.
Yeah, to again, whether it's nostalgia or whatever it is, absolutely that, or just make you hungry so that you want to eat, or any of those types of things, absolutely. Right. Notice also associated with this sense of smell, associated with our memory, are two other important structures that we need to talk about. The first of these is this round structure down here known as the mammillary body. Now, why am I talking about the mammillary body? I don't see it here on the list in the limbic system. So why am I talking about the mammillary body? Is it something you need to know?
Yes.
Okay, but it's not here in the limbic system, so where is it?
I thought it was part of the what not thalamus, but what was it? I just did the lab on it.
There you go, the diencephalon. That region of the brain. Is it on your second page of the handout? What number is it under the diencephalon?
Yeah, it's number 49.
There you go, number 49. Mammillary body. Excellent. Turns out this mammillary body, which indeed is a part of the diencephalon, it's not a part of the telencephalon, but we will talk about it right now. The mammillary body is what is responsible for taste and smell aversion, right? If you're a sheep and you eat a red berry that makes you sick, do you want to eat that red berry again? Come on, these are the easy answers.
No.
No, of course not. So you're going to remember the smell of it. You're going to remember the taste of it. So if you ever taste something like that tastes like that again, you'll spit it out. Or maybe you're not a sheep, maybe you're my wife and you're pregnant with our first child and living in Southern California. And there's a restaurant there that has this amazing black pepper ranch dressing that they use on their salad, which you love so much that when you're pregnant, you typically eat there at least once a week. Until one night you end up, because of the being pregnant, getting sick, throwing up all over the place after eating there. And did my wife ever go back to that restaurant and ever eat that peppercorn ranch dressing ever again? Never.
No.
Absolutely. Because of that aversion of it, because of the taste associated with the getting sick. And that's what it does. That's what it's responsible for. And the reason we're talking about it here is, notice there are some axons. That's what this yellow bundle is. These axons that basically carry information from the mammillary body to the hippocampus. And this bundle of axons is a structure known as the fornix. We see it here in this nice three-dimensional view, but notice in the sagittal section, we see it too. We see it here, and we can tell because of the color of the yellow, but also notice here, we can tell this is it right here. This structure here is the fornix. Well, it depends. It is the fornix. And again, it can easily be seen. And here we see that it is white matter. Now, notice it is not the only white matter structure that we see in this sagittal section. By far, the biggest white matter structure is this big comma-shaped structure known as the corpus callosum. Again, these are white because they're made of axons. And this corpus callosum is the largest connection of commissural axons. Remember, as we talked about, commissural axons are the ones that go from, where is it? Here we go. The ones that go from one hemisphere to another. So these axons carrying information from one hemisphere to another are what is known as the corpus callosum. So here we see it from a coronal section. And like I said, here in a sagittal section, we see it as well. So this is the corpus callosum. And notice that corpus callosum right below it, we can find the fornix as well. Now, I believe it was in the last class, someone was talking about epilepsy. I don't remember who somebody typed or asked about, but we were talking about.
That was me.
Okay, excellent. We were talking about epilepsy, and we mentioned how, and actually let's go to this picture. Epilepsy is a condition where individuals get inappropriate firing of neurons, and they produce this big, huge, what they call neural storm, where all of these action potentials are produced by all these neurons, and it can spread across the entire surface of the brain. It can start typically in one location, but it spreads along the entire hemisphere, and because of the corpus callosum, it can spread to the other hemisphere as well. When this occurs, these individuals typically have what we call grand mal seizures. They drop to the ground, convulsing. This is when you got to put the spoon in their mouth so that they don't bite their tongue off. These people have a very challenging time leading a normal life, let alone having difficulty sitting in a classroom or driving a car or doing all the other normal things we take for granted. However, as was also mentioned in the last class, one of the things they can do when these seizures are really severe is they can actually make an incision through the corpus callosum, separating the two hemispheres of the brain. Now, again, there are still some other commercial connections, but this is by far the largest. And so now when that neural storm is produced, it's not able to spread to the other hemisphere, and it can dramatically decrease the severity of the symptoms. And for some individuals, they're actually then able to live a relatively normal life. Of course, as normal of a life as you can have with the two sides of your brain not being able to talk to each other, right? If the two sides of your brain can't talk to each other, that's got to be pretty debilitating, right? Well, yeah. Turns out it isn't. People who have this split brain surgery, often it is very difficult to tease out the deficiency. Why is that? Well, because if you see a can of soda, a real live object in front of you with both your right eye and the left eye, can your right eye tell where it is? Can your left eye tell where it is? Can you reach out with your hand and grab it? Yeah.
But wouldn't you have trouble like passing it on from one side?
True, you would have. They could have problems passing it from one hand to another. So two-handed coordination of things can be challenging. Although they can still be pretty good playing piano, because again, often when you're playing piano, the two hands are doing different things. But often, so yes, there are some deficiencies that individuals have, but many of the deficiencies from the split brain are relatively negligible. There are some ways they can tease them out, and these things are really cool. One of the things that they'll do is they'll give them a special viewer that they look in, and they're presented with an image of a face. And what'll happen is on one side it is female, and on the other side it is male. And so what they will do is they will tell them to write down what they see and say out loud what they see. And they'll write down male, and they will say female, but they won't realize that there's a difference in what they've said and what they've written. They won't be aware of the difference in there. But obviously, most people that walk around the world aren't male on one side and female on the other. So again, it can be surprisingly functional. Now, often people that have epilepsy have other problems or other neurological difficulties. So again, they can't necessarily live normal lives, but like I said, the problem from splitting that corpus callosum is not nearly as devastating as you might think.
Wouldn't you be better at multitasking though, since you don't have that connection
Possibly.
between brains?
Yeah, so maybe a better piano player because of that, or some other type of instrument where you don't have to necessarily, where the two hands are doing different things. Lori, you had a question?
Yes, there was a question on the check your understanding part where it states that agenesis of the corpus callosum, the lack of development of it, does that kind of tie hand in hand with the seizures here? Like right here, you're splitting the left and the right hemisphere, but in that defect, it's the lack of development of the corpus callosum.
Well, so if the corpus callosum doesn't develop, my guess is that they would have less connections in between. And so it would be just like splitting it. Yes. So it would mean that they would be less likely to have it spread from one side to another. So they would have less severe seizures, would be my guess.
Okay.
Yep. Excellent. All right. I want you to notice one other thing as well. Well, actually, I want you to notice many things that we're talking about. Notice with this particular view here, here's our corpus callosum. And notice underneath it is a little bit of tissue as well, some nervous tissue here between two spaces. There are actually spaces inside of our brain known as the ventricles that contain cerebral spinal fluid. And these two that are to the side are known as the lateral ventricles. Notice if instead of a coronal section, we take this kind of weird transverse section that we see here, we can see one of these ventricles is this big C-shaped space on one side and one on the other, but the lateral ventricles are separated from each other. They don't directly connect. There's a little bit of tissue that separates them, and that little piece of tissue that separates them is the septum pellucidum. So here we see the septum pellucidum from a top view. Notice it is underneath the corpus callosum. So if we look again at this picture, here is the septum pellucidum beneath the corpus callosum. But the other place that you see it that makes it really easy to find, actually, let's even use that picture. I like this one even more. Corpus callosum, fornix, and notice in between the corpus callosum and the fornix, this little piece of tissue right here, that is the septum pellucidum. So find the corpus callosum, find the fornix, and that's the corpus callosum. Notice if we were to poke through that with a stick, basically we would be in the lateral ventricle, right? Because basically what we would be doing, see, and here we see it as well. This is the septum pellucidum. We would basically, this is that wall we were looking at. So notice if we were to poke through that wall, we would go into the lateral ventricle from the other side. So that's what we're seeing when we look at that. So again, that's a structure, doesn't have a function other than, I guess, forming the wall of the lateral ventricle. So it doesn't have a structure that way that we need to necessarily be able to identify. What else do we have? Oops, I moved on to the diencephalon. So let me go back up here. We did that, we did that, we did that. Is it just the basal ganglia we have left?
Yeah.
Excellent. Excellent. Let's go back. Let's go back to this picture. All right. As we've learned many, many times, anatomists hate us. And again, one of those things comes in the form of definitions. Remember, as we've talked about, a collection of neuron cell bodies in the central nervous system we call a nucleus. And of course, the plural of that is nuclei. A collection of neuron cell bodies in the peripheral nervous system is known as a ganglion. So the plural being ganglia. Excellent. Well, notice we have here in the middle of our brain not one, not two, but three clusters of cell bodies, all located together here deep in our cerebrum. And so being a collection of cell bodies inside of the central nervous system, they collectively are known as the basal ganglia. Is that right?
No.
No, it is not right, but it is how they are most commonly referred. Now, if you look in the literature, it is getting better. More and more people are now referring to it as the basal nuclei. But it is still in the literature surprising how often you still see the term basal ganglia. So even though basal ganglia isn't a technically accurate term, it is used so frequently, I will accept both. So on the exam, if I show you this picture, I have that circle, I ask you to identify these three things collectively, you will call them the basal ganglia. Now notice it's made up of three things called the caudate, the putamen, and the globus pallidus. But I don't see those names on my list. Do you see those names on your list?
No.
No. So much like the muscle groups from the last one, you don't have to know those individual names. You don't have to know the individual names of these three nuclei. You just need to know two things. Collectively, they are known as the basal nuclei, and you need to know what they do. This basal nuclei is essentially your autopilot. Now, what do I mean by that? Well, think back, or not think back, because hopefully most of you don't remember it, but think back to when you're a kid or you saw a baby, a 1-year-old, a 10-month-old trying to walk. When they're trying to walk, you can almost see the steam coming off of their heads. They're putting so much thought into where every foot is going and where their arms are going and how their leg is moving. They have to think everything about that because they are learning that behavior. Does it require that much thought for you to walk? No. While you're walking, you're talking to your friends and you're tweeting a picture and you're eating a sandwich and you're doing your makeup and you're doing all of these things. It is a learned behavior, right? Or driving a car. When you first learned how to drive a car, right? There was like a cobra that was sitting on the dashboard because you were paying attention to everything and your hands were always at 10 and 2, right? But now it's a learned behavior. So you're doing your makeup and eating a sandwich and you're tweeting. And the problem with that autopilot is sometimes it kicks in a little too hard because you got in the car to go to the store to buy milk and you're halfway to work before you realize, oh crap, I'm halfway to work. Or in the shower and you can't remember whether you washed your hair or not because your autopilot kicked in, right? This basal ganglia helps us to maintain a learned behavior. My favorite example of this is actually my wife when she was trying to learn to knit. Her grandmother is an amazing knitter. And one summer when we were visiting her, she tried to teach my wife how to knit. And grandma is one of those people who has her knitting needles and she can be sitting there watching TV, having a conversation, making a pie, all while she's whipping out that sweater, right? She's not even paying attention to what she's doing, but she's doing it a million miles an hour. Whereas my wife had to think about every knit and every purl, right? Much more concentration. Whereas grandma was using her basal ganglia to just keep that learned behavior going. All right. Questions on that? All righty. I've been talking for a while. You guys are getting quieter. I think that may mean you're tired. Let's go ahead and take one last break. We'll take a 15-minute break. It looks like it's 3:07 now. So let's continue at, what will that be? 3:22. We will start back up in 15 minutes. Give you a chance to get a quick stretch. This is actually much more straightforward than the muscle system. With the muscle system, you had to learn a muscle, you had to learn its origin, you had to learn its insertion, you had to learn its actions. Here, you just have area, function, area, function, area, function. It's very straightforward. It's not as scary as it seems. Yeah, exactly. That's good. The vast majority is, what is this area? What is the function of this area? Right, that's pretty much most of what this exam is going to be, or a lot of it anyway. Identification and function. It's all the labs exams are. All right, excellent. So take a break, come back 15 minutes, and we will do some more anatomy. All righty, excellent. So we've left off and we have worked our way through all of the telencephalon. I know we still need to talk about the flow of cerebral spinal fluid and the protections, but we'll do that probably in the next class when we get to that. So I want to move instead down to the diencephalon. On this simple illustration that we saw previously, the diencephalon is just this kind of oval shape looking structure here. However, I think where you see it best, well, here, and they've peeled away all that pesky cerebrum so that we can see it. But where I think we see it the best is here. Oops, where am I going? Here with a sagittal section. With the sagittal section, we can see that fornix, which is basically the top of it, and then this narrow neck region, which is the mesencephalon. So basically our diencephalon is this region in here. Basically, it is all of this stuff. And to me, it always kind of looks in this sagittal view. I'll remove all my drawings so you can see it. It kind of looks like the head of a duck. We got a little bit of the bill here, and we got this funky looking eyeball. All right, so it kind of always looks that way to me as like a head of a duck. Now, the most prominent structure of the diencephalon is actually this structure right here, this bulging structure out here known as the thalamus. If we go back a previous picture, here we see the thalamus sitting on top there, and here we've pulled it out and see what the thalamus is. The thalamus is actually a collection of nuclei. So again, remember, nuclei are clusters of cell bodies, and these are basically the relay station for most sensory information. Notice I didn't say all, because remember, our sense of smell goes straight to the limbic system. But pretty much every other sense comes here to be processed and to move from here to the parts of the cerebrum for processing. So as we talked about, our sense of touch goes to the posterior central gyrus, our primary somatosensory cortex. Our sense of sound goes to our primary auditory cortex. And our sense of vision, remember, as we talked about, is going to go back here to that area 17 primary visual cortex. And notice we see all that stuff. In fact, notice this little green nuclei right here is known as the lateral geniculate nucleus. This is the one that receives our visual input. Now, if you'll notice, that term lateral geniculate nucleus is not on your list. However, that is where I did most of my research that got me my PhD. And my grant requires me to say the term lateral geniculate nucleus at least 3 times in this class. And so now I have. So excellent. We have done that, and I get to keep my money. But the point is, the thalamus is a relay station where sensory comes in, and then it goes to the area. No, you do not have to know those Brodmann numbers. Remember, I just used them the same way if you were looking at a model of the arm and I said the bicep brachia was number 14. You could find number 14 on the arm and be able to easily see what muscle I was talking about. So I'm just using it strictly as a label. It is not definitely not anything you need to know. So the thalamus is a relay station. Sensory information comes up to the thalamus, and then from there goes to whatever region of the brain is necessary. All right. Notice it is here. And we have two thalami, but the two thalami don't touch. There is actually a space in between them. In fact, I think I have a view of this. Do we not have a view of this? I think I have to go back to that previous picture. Here we go. Excellent. Here we go. This is perfect. This is the view I wanted. Let's actually do that. Excellent. Notice if we look at this picture here, we can see two very important things. The first is this thalamus on this side and this thalamus on that side, the two thalami, with a space in between. And that space in between, which I will draw as blue, is our third ventricle. There is a fluid-filled space between them. However, if you look closely in that space, you will notice that while the two thalami don't touch, there is a tiny bundle of axons that connect the two. That tiny bundle of axons that connect the two are what are known as the intermediate mass. So notice if we do a sagittal section and cut this in half, and then look at it from the side view, lo and behold, this little structure right there, that little bit of white, is that intermediate mass, that cut through bundle of axons that connects the two thalami together. So that is the thalamus, that is the intermediate mass. And this space is the third ventricle. However, obviously that space has a wall that makes it. And if we look at this wall looks solid and smooth and uniform. But if we take a closer look at it, what we see is in that smooth surface underneath the thalamus is a big bundle of nuclei. And this big bundle of nuclei located right there below the thalamus is known as the hypothalamus. The hypothalamus plays a very important role in helping to regulate many important functions of the body. Thirst helps to maintain blood volume, blood pressure, plays a role in heart rate, plays a role in lots of different basic physiological processes. But one of the most interesting things that it does is it controls this little dangling M&M that sits beneath it. This little dangling M&M that sits beneath it is what is known as the pituitary gland. The pituitary gland is our master endocrine gland. It produces hormones like thyroid stimulating hormone. Guess what thyroid stimulating hormone does?
Stimulates the thyroid.
Exactly, absolutely, right? So it controls most of the other endocrine organs. And in fact, for many years, they thought the pituitary gland was the command center for the endocrine system. It produces over a dozen hormones that control most of the other endocrine glands. But what they found out only like 20 years ago, which is a relatively recent finding, is that this hypothalamus actually controls the pituitary gland. So the pituitary gland is the figurehead, right? It's the one there out in front, but the hypothalamus is really the power behind, the control behind the scenes, right? Pituitary gland is the president, right? The hypothalamus is the Illuminati, protecting, leading from the shadows. And that's kind of what it does here. And notice that M&M that dangles down there connects by a little stem known as the infundibulum. We've talked about this pituitary gland once before. Go ahead and clear that. Where'd it go? Oh, I'm all over the place. I want this picture again. There we go. Remember when we talked about the bones, this U-shaped indentation, the sella turcica of the sphenoid bone. Remember, we also said that it was the hypophyseal fossa. It was the indentation for the hypophysis, which is a fancy name for the pituitary. So notice here's the pituitary gland, here's the infundibulum, here's the hypothalamus, here's the thalamus, and there's our intermediate mass. And notice right down here below it is that mammillary body that we talked about that communicates via the fornix to the hypothalamus. Now I keep calling it the mammillary gland, and obviously it has nothing to do with the mammillaries. It has everything to do with our sense of smell. So it may seem like a weird name for it, but notice if we look at an inferior view of the brainstem, notice here we can see this is where the pituitary gland would sit, and you see these two globular nuclei side by side that are known as the mammillary bodies because they look like two little tiny mammillary glands on the bottom of the brain. Right? Again, as I've said many times, our anatomists are very sad, depraved, lonely individuals. Thought I had that one picture of the brain. Where did it go? There we go. So again, notice here is, here's the infundibulum. So again, our pituitary gland would sit right there. And there are the two little mammillary bodies. Excellent. All righty. What's next? Oh, perfect. Excellent. All right. So we come back to this picture. We have our infundibulum. We have our pituitary gland. And notice there's this little white matter in front of us. This little white matter in front of us goes back to this image we were talking about with our eyeballs. Remember, we have this interesting condition where we see information from both the left sides of our visual world. So some of the information needs to stay on the same side and some of the information crosses. So we, on the left side of our brain, we have everything from the right side of the body visually. And on the right side, we have everything from the left side. So we have names for all of these bundles of axons. The bundles of axons that come out of the eyeballs, not surprisingly, are known as the optic nerves. It's our second cranial nerve. However, they come together into a single structure where some of the axons cross and some don't. That is known as the optic chiasm. From there, once we get that information crossed, all the stuff on the left travels in the on the right optic tract. All the stuff from the right travels on the left optic tracts, and they go to the thalamus. Notice in the thalamus, as we mentioned, specifically that lateral geniculate nucleus of the thalamus. Hey, I got to say it a fourth time. Lucky me. And then from there, it goes to our primary visual cortex. We see this in this nice illustration. We see this, come on, in this nice illustration that we saw as well. Here, since we're crossing through the midline, we are cutting through the optic chiasm right there. Here we see the same thing. Here we see the two optic nerves. And what's the picture that I wanted? Oh, there we go. This is the last one I wanted. Notice here we see where the two optic nerves come into the optic chiasm, and then the optic tracts leave from there. So it's this X-shaped bundle of axons that we are seeing. Now, the last picture I want to do is come back to this one here, because one of the things that I wanted to mention is, let's say, for instance, you happen to have a problem with your pituitary gland. And actually, let's go back to this picture. Your pituitary gland starts to swell. As it swells, it is going to expand upward, and it pushes on the optic chiasm, where some of those axons split. And what's going to happen is you're going to get a total tunneling of your vision. Suddenly, you're only going to be able to see right here. You're not going to be able to see anything over there. You, of course, go rushing to your optometrist. And is there anything your optometrist can do for you in that situation? No, of course not. Well, for the most part, optometrists are worthless individuals. But for the other reason is this is obviously an endocrine problem and not a problem with your eyes or anything along those lines. So yeah, but it gives you a sense of if there's damage to that area, the parts that cross get damaged. And so you get this tunneling of your vision. So those three structures are all just bundles of axons carrying different types of information. And an easy way to see its function would be to damage it. Notice if you cut an optic nerve, that'd be the same thing as ripping out your eyeball. If you ripped out your eyeball, how would that affect your vision?
You wouldn't be able to see out of
Well, from
that
your right eye, yeah. It'd be the exact same thing as if you winked. Close your right eyelid. If you close your right eyelid, you only see the world with your left eye. And that would be what it would be like if you damaged your optic nerve. As I mentioned, if you had damage to your optic chiasm, you would get a closing, a tunneling of your vision. But what's really cool, well, not cool to hurt the brain at all, but does the concept of it. If you were to damage an optic tract, if you were to damage the right optic tract, what would happen is you would be able to see stuff out of both eyes, but all you would see from both eyes is the right side of the world. So basically anything over here from the left side of the world, I would not be able to see no matter which eye I used. If I use my right eye, I'd see this side of the world. If I use my left eye, I'd see this side of the world, and that would be it. So this contains eye-specific information. This contains the crossing axons. And the optic tract contains a side-specific information. All right, there you go. Excellent. Couple more things. Remember, we talked about how the corpus callosum is the largest commissural tract, the largest bundles of axons carrying information from one side of the brain to the other. But we've learned there are others. One of them was the intermediate mass. But there are two other ones as well. This here in pink and this here in green. Notice one of these is in front, one of these is in back. So guess what we call this bundle of axons that goes from the left hemisphere to the right hemisphere in the front?
Anterior.
Anterior what?
Measure.
Nope. Anterior bundle of axons that goes from one hemisphere to the other. What would we call it? If only I had a list of terms in front of me I was responsible for. Maybe 47?
The commissure?
Yeah, anterior commissure. Excellent. And what then would the green one be?
The posterior commissure.
There you go. See how easy that was? They're pretty easy to find. In particular, the anterior commissure, because if you notice, the fornix points right at the anterior commissure. We see this here in the illustration. We see it in the real brain. I also want to go back to that chart picture. Here in our chart picture, notice the same way. The fornix points right at the anterior commissure. Now notice it's axons, it's white matter, but I think they made it black here just to make it stand out and show that it's different. And then of course, right across the third ventricle from it is the posterior commissure. You could also follow the corpus callosum. Notice the corpus callosum goes straight to the anterior commissure. However, our corpus callosum doesn't go straight to the posterior commissure, because if you see, there is something else in the way back here. This structure back here in the way is what is often referred to as your first chakra. It is often referred to as your third eye. What else is this structure known as?
Is that the pineal body?
That is the pineal body or the pineal gland. Absolutely. Both are acceptable terms for that. What does that pineal body or the pineal gland do? Anyone know? I know. What does the pineal gland do? It's a gland, so it probably makes something.
Is that where melatonin comes from?
Excellent. Melatonin. Right, melatonin. Oops, helps if I spell it right. Melatonin. Melatonin, that's the stuff that makes your skin dark, right?
No.
What makes your
No,
skin
it's for sleep.
Not melanin.
Melanin.
Oh,
makes your
melanin, that's
skin.
right. I always get those two confused. So what you guys were just telling me, what is it that melatonin does?
Sleep and wake cycle.
Yeah, absolutely. This plays a huge role in our sleep cycle. Absolutely, right?
I don't think my mom has that structure because she never sleeps.
Possibly, or it's been disrupted. Absolutely, that can be one of the issues, and we'll talk about some things that can cause that as well. So, absolutely, right? We actually have our own circadian rhythm. Again, remember those freshmen in the psychology classes that we talked about that got tortured? This is one of the ways we found that out. Because I don't know about you, but how many people in this class right now woke up this morning because they wanted to? Woke up on your own, just smiling and stretching and happy to be awake?
I woke
Anybody?
up on my own, but yeah. I
Yes,
went
you
back
did?
to sleep.
Okay, well, you went back to sleep. You weren't that happy to be up. So for
Yeah, I
most
wasn't.
of us, no, we have kids, we have responsibility, we have jobs. We get up because we have to. We eat when it fits into our schedule, right? We have a tremendous number of social cues that determine when we do things. Yeah, like when you're a 9-year-old in the summer, you get to set your own schedule, right? But obviously, birds know when to eat, they know when to sleep, and they don't even have watches. So what they were curious about is, are humans able to basically tell time without social cues? So they took a bunch of freshmen and they locked them individually into rooms in a basement. These rooms had no windows, right? They had lights that could be controlled, turned on and off, no clocks, no televisions, nothing that would allow them to perceive time. And they left them in there for a little over a week. And what they found is, after a couple days of acclimating, a student would tend to get up at the same time every morning. They would tend to eat at the same time every day. They would tend to go to sleep at the same time every evening. Now, again, not every student got up at 7 a.m., right? 8 at 9 or whatever, but each individual student kind of ended up on their own fairly stable schedule. And as we learned, the reason for that is this hormone melatonin. Melatonin basically helps to make us sleepy. And as during the course of the day, we build up melatonin levels so that by the end of the day, we are tired as a result of that. Yes, Laura.
Yeah. Do are melatonin gummies kind of like not
The
useful?
worst thing that you could do for yourself? Yes, absolutely. Right? Because here's the deal. Melatonin makes you sleepy. Absolutely. And so as you build it up, you make it, as your pineal gland produces it during the course of the day, it makes you sleepy and it makes you go to bed. But like all hormones, we regulate hormone levels with a negative feedback process. So tonight you have trouble sleeping, so you take a melatonin pill or a melatonin gummy or something along those lines. What that does is it spikes the level of melatonin in your body, making you more tired, making it a little easier for you to sleep. But what your pineal gland sees is, holy crap, there's a ton of melatonin in the body. I have to stop making it. And so it suppresses the amount of melatonin that your pineal gland makes. So the next day, it's been suppressed and it makes a little less. And so that night, because it's making less melatonin, you take another melatonin pill, jacking up your melatonin levels, suppressing your pineal gland even more, until it gets to the point where your pineal gland practically can't make melatonin anymore. And if it's not able to make sufficient amounts of melatonin, are you ever going to be able to come off that pill? No. So spiking the levels of the melatonin can be very dangerous for the normal function. So please, I encourage you not to do that. Just sleep. True. The CBD is supposed to be very calming, can affect things that way. There are other things that you can do to calm yourself to try to find ways to get to sleep. I'm not sure why melatonin would help your, I guess that if it makes them more sleepy, but again, that would be a concern. I would say I would not encourage that. All right. Yeah, I'm not a medical doctor, so obviously listen to your medical doctors. But from an anatomist standpoint, and or understanding anatomy and physiology, you have to be very, very careful with those types of supplements because they can disrupt your normal body's ability to make those. And with a 2-year-old, I personally wouldn't want to do anything with my kids that would disrupt their normal production of hormones. All righty. I think that's it. Is that it for the diencephalon? Did I miss anything? I don't think I did.
I don't recall talking about infundibulum. Did we talk about the
infundibulum, remember, is the stem that holds the pituitary gland to the hypothalamus.
Okay, thank you.
So yeah, so let's look at a better picture. Yeah, there we go. So this stem that holds the pituitary gland to the hypothalamus, that's the infundibulum. Here we see it cut. All right. Mammary bodies, remember, are these structures here that give us, those are the ones that did the smell and taste diversion that we talked about. All right. Excellent to do all of that. Perfect. Excellent. Yeah, we talked about that one in the telencephalon because remember, it's the thing that the fornix connects to. So that was where we talked to it there. Where are we time-wise? Okay, we're still doing okay. All right, excellent. So the next region is this very narrow region indicated in green, known as the mesencephalon. The mesencephalon is also sometimes referred to as the midbrain, so you can see it both of those ways. From an anterior view, like we see here, the most prominent features, no, it's not going to work for that. The most prominent features are these big, large knuckle-like structures. And these big, large knuckle-like structures are massive bundles of axons. Because after all, a tremendous amount of information has to go to and from your brain as your brain is processing and making sense of information. And these big, huge, massive bundles of axons are known as simply the cerebral peduncles. I have no idea what a peduncle is, but whatever it is, it's a large bundle of axons going to the cerebrum. They're big and prominent on the front of the mesencephalon. But I think one of the easiest ways to see them is if we take a cross section. If we take a cross section through the mesencephalon, what we see is something that looks like this. And notice now we see this big, huge bundle of white matter. That is that cerebral peduncle. All right. And again, It's just a bundle of axons carrying information to and from the brain. Seedweed are some of the protections around the brain and spinal cord that we'll talk about in the next class. Deep to the cerebral peduncles, you may notice, and I'm just going to draw around this so you can really see it better. There is this darkly pigmented region. This darkly pigmented region is very dark in color, which is where it gets its name. All right, it's the dark substance. It's the substantia nigra. This substantia nigra is where our friend that we were talking about before, dopamine, is made. Remember, we talked about dopamine as we are finding more and more plays a role in social anxieties and things along those lines. But we also talked about it in terms of Parkinson's. Remember, in individuals who have Parkinson's disease, they don't make their own dopamine anymore. And so this region in their brains is completely clear. There is no dark pigment to it at all. This becomes a completely clear area. And this clearness to the substantia nigra is actually how they diagnose Parkinson's. Parkinson's in a living individual is pretty much a diagnosis of symptoms. We see the symptoms. Those symptoms have a particular classification to them. And so it's indicative of Parkinson's. But it isn't till they cut their midbrain and actually look in there and see that there's no dopamine that they're actually able to confirm the diagnosis of Parkinson's. Now, obviously, this is not something most people are willing to have done while they're alive. So usually a diagnosis of Parkinson's isn't actually confirmed until after the individual dies. All right, a couple more things. Yeah, dopamine is an important hormone, as we mentioned, plays a role in that waking, the still tremors, the inactivity of movements. And like I said, a lot of recent studies have shown dopamine plays a role in a lot of social anxiety interaction type of disorders. Yes, question.
Yeah. For the cerebral peduncle, you said massive bundle of axons that carry axons from where to where?
Spinal cord up to the brain, to and from the brain.
Okay.
Yep. So dopamine is more of a neurotransmitter. Again, it is a chemical signal, so it can act like a hormone, but hormones typically circulate in the blood, whereas neurotransmitters are typically released by neurons. So I think it'd be more accurate to say dopamine is a neurotransmitter, although it can have some hormone-like functions. You know, hormones are one of those things that are tricky to study because, again, it's hard to see these cells working at a cellular and chemical level in a living individual. So it can be challenging to understand, but it's definitely a neurotransmitter and seems to have some hormone-like interactions. Yes, question.
Yeah, so for some reason I was under the impression that hormones are made only in glands. So is this a gland?
No, because like I said, it is considered a neurotransmitter that may have some hormone-like properties. So this is not considered a gland. So no, it's the neurotransmitter.
Clarification, thank you.
Yep. All right, it can't be seen quite as well on this cross section. So let's cheat and look at a pretty picture. Now, for whatever reason, notice this pretty picture is upside down what we were looking at before, but we can still see the big knuckle cerebral peduncles. We can still see the substantia nigra. But notice, and I'll change colors, this fuzzy region right around here. This fuzzy region right around here, there's a little hole where our cerebral spinal fluid flows through called the cerebral aqueduct, which we'll talk about later. But these two shaded regions are what are known as the reticular formation. We were talking about the role that your pineal gland plays in being fatigued, wanting to sleep, going to sleep. However, sleep is a funny thing, right? When my wife, for instance, was sleeping, the dog could be barking, right? Sirens could be going off. Machine guns could be going on outside, right? Construction workers could be doing their work, and she will soundly sleep through all of it. But when our first child whimpered just the teeniest bit, what would happen?
She would wake up.
Instantly awake to take care of it, right? Or when we were first married, she would fall asleep, would be snoring very soundly. I would get up to go play Xbox, and just that creak of the floorboard, she was instantly awake, right? No, you don't get to get fun, get back in here in bed, right? That ability, obviously, it's not the amplitude of these sounds that are causing her to wake up, it is the significance of them. And that's what this reticular formation is. This reticular formation is kind of that alert system. Different sounds are going to have different significances to us. You hear stories all the time about siblings who sleep in the same room. And one of the siblings could be throwing a temper tantrum and screaming and yelling and wanting their mommy, and the other one is just snoring away, completely oblivious. Right? While those noises are loud, they're not significant, they're not important, and so we're able to ignore them. Whereas much more significant sounds make us much more alert. That creaking of the floorboard, that whimpering of the baby, those types of smaller, even subtler sounds are something that we're much, much more aware of. Right? Now, this reticular formation can be stimulated by other things as well. One of the things that they always warn you about if you're having trouble sleeping is the blue light, for instance, of your phones or of your television. This is where that occurs. Your reticular formation can be stimulated by light, especially more bluish light. And when this area is active, you can have a harder time falling asleep. So again, light can be an issue. It's one of the reasons why most night lights have changed in the way that they were. Back in ancient times, and by ancient times when I was a kid, a night light was this little light bulb you plugged into the wall socket. You turned it on at night and you turned it off in the morning. And what they have found is that while it's great for keeping the boogeyman away, if there's too much ambient light in the room, the child may not be able to get full deep sleep. And so they can have trouble with sleeping that way. So now most night lights have a timer on them. They're on for 15, 20 minutes, long enough for the kid to fall asleep. And then they turn off so you don't have that ambient light, and they're more likely to get deeper, more restful sleep as a result of it. Wow, how big is a giant spider? It's got to be pretty large if it's making a noise. Excellent. All right. Yulia, did you have another question, or was that the one from before?
No, that's an old one. Sorry.
No problem. Excellent. All right. And I think that oh no, two more things. Excellent. Notice on the posterior, so on the anterior part, we have those big, huge knuckle-like cerebral peduncles. Exactly. On the posterior part of the mesencephalon, we have these 4 large nuclei. Collectively, these 4 large nuclei are known as the corpora quadramina, right? Quadramina, 4, nuclei basically is what that means. I don't know what corpora means, head or something like that, but there are these 4 structures collectively, the corpora quadramina. However, notice 2 of these are on top and 2 of these are on the bottom. So the 2 on top are what are known as the superior colliculi, and the 2 that are on the bottom are known as the inferior colliculi. These colliculi serve a very important function. They help you to find your beer, right? Because there's your beer sitting on the counter, and it seems like it would be something as simple as the light from the can hits your eyeball and allows you to know where it is in space. But here's the problem with eyeballs. Eyeballs can be moved, right? The eyes themselves can move left, right, up, and down. And depending on how I move my eyes, the light from that can is going to hit my eye in a different place. Plus, my head can move. Plus, my body can move. So even though light is falling on my eyeball, to really understand where something is in space, so I can reach out and grab, again, it's not a beer, it's a Coke, but to grab that can of beer or whatever else you're drinking, or to see where anything is in space, you need to be able to combine two pieces of information. You need to be able to combine the light, what we are seeing, and the position of your body, head, and eyes. So basically, we have to combine our visual system and what we call knowing where your body in space is a perception we call proprioception. Proprioception is why when your eyes are closed, you can still touch your nose with your fingers. You know where your body is in space. You don't fall over when you close your eyes because you lose your balance and fall. Unless you're drunk, which can impair your sense of proprioception and make it harder for you to touch your nose with your eyes closed. But for the most part, as long as you're sober, you have a pretty good idea of knowing where your body is in space. And that is what our superior colliculus does. Our superior colliculus takes our body's position and where light is so that we truly know where objects are in the world around us. And while I can't move my ears the same way I can move my eyes, I can move my head, I can move my body. So the inferior colliculus does the exact same thing for sound and position of your body and head, or again, combines auditory information and proprioception. Now, this is the point in the class where I've usually already spoken with somebody who sits in the back of the class. And at this moment, as soon as I say that and finish writing this on the board, they take their textbook and they slam it on the ground. And if we were in the classroom and someone in the back of the class slammed their book to the ground, what would everybody do?
Look that way.
We'd all turn and face it. And the whole point of turning and facing it is you take all these senses we're talking about, where your body is in space, where your eyes are, where your ears are, and you focus all of them on that sound to see and perceive if it's a danger, if it's a threat. That's what this Scapora quadramina does. It allows us to combine all this information to allow us to be able to assess a threat. So if there's suddenly a big sound in the back of the room, we can all rotate to it, coordinate to it, and make sure it's not a bear with an axe trying to come into the room. Nope, it's just Laura dropping her book like I asked her to. All right. Questions on that? All right, excellent. That brings us through our mesencephalon. All we have left is the met and myencephalon. It is still a little bit early, but I know I've covered a lot of material. So I think we are doing okay where we can finish this and start the protections and start the spinal cord on the next class and still be okay. We are going to start those things, and we are still going to try to get the cranial nerves done on the 27th as well. So make sure you have that handout or something similar to that handout. Your book, the section in your book where it talks about the cranial nerves, does an amazing job of talking about it. So I would strongly encourage you over the weekend to look at that ahead of time. So like today, you're not overwhelmed with the massive amount of information we're talking about. Even if you don't understand it all, if you take an opportunity to look at it first, I think it will help you tremendously when we talk about it on Tuesday. So I'd strongly, strongly, strongly encourage you to do that. All right. This is a good natural stopping point, so we'll go ahead and stop here. Any questions before we call it a day?
I have a question. Are we going to mostly have illustrations or like cadaver pictures or a combination of both on the lab?
So for this lab exam, while there will be some real pictures used and even pictures of models, right? The advantage of having pictures like something like this, or even something like this, the advantage of these things is the color coding can be tremendously helpful, right? Again, your job in this class is to basically gain and understand this information. If I just gave you a real brain, assuming it didn't have this label on it, with enough time and effort, you could probably find from the top of this the brain the central sulcus and follow it down towards the lateral sulcus. You'd be able to find that. It would take you some time, but you could probably do it. However, if I give you a picture like this, is it going to be a lot easier to find the central sulcus?
Yes.
Absolutely. And since the point of this is for me to test your knowledge and understanding of this information, I don't have a problem with using obvious examples of these things. Yes, I guarantee there will be pictures of some models, pictures of charts. I've shown you that chart a lot of times. There may be, again, yes, when you look at a brain, an actual brain, like this one, right? There obviously can be some intimidating things to look at. I'm not going to have you try to figure out where, obviously this isn't the view where you would see it anyway, Broca's area is or something like that on that. But there's plenty of really, really obvious stuff. The olfactory bulb and the olfactory tract, the optic nerves, the mammillary bodies. This structure here we haven't talked about yet, but it's the pons. This structure here is the cerebellum. So there are some big structures that absolutely, some obvious structures that absolutely you should know and be able to recognize from something like this. But am I going to be pointing at this little gyrus and ask you to identify what that specific function of that gyrus is on this one here? No. But yeah, I will use pictures like this as long as it's something that I think is really, really obvious.
Okay, thank you.
Again,
it just kind of helps for studying to know.
I totally appreciate that. And I think it was something I tried to emphasize on the last test. If you think about the last lab exam, I told you guys exactly what was going to be on the lab exam. Illustrations, pictures of models, pictures of bones, and some histology, and some bodybuilder pictures. I told you exactly what was going to be on it, right? And yet still, many of you did not do as well as you probably should have on that. So again, my goal isn't to be tricky. I appreciate how hard this is. So I always try to use obvious examples, and I have no problem telling you what I think is a good picture and what's not a good picture and things along those lines for doing it. But at the same time, I'm not going to say memorize this picture, right? This picture is a great view of the thalamus, but this is not the only great view of the thalamus. So I'm not going to say this is the one picture we're going to use for the exam, memorize this, because that's not real knowledge. If you can recognize the thalamus on this, but also the thalamus on the chart, and also the thalamus somewhere else, then you truly understand the thalamus. If you just memorize this duck eyeball equals thalamus, then that, in this particular picture, then that doesn't necessarily meet the requirement of what we're looking for in this class. So I'm not going to say these are the 10 pictures you need to memorize for the exam, but is this picture likely to be on the exam? Yeah, absolutely. But others like it will be as well. All right, awesome question. Any others? All right, excellent. All right, I'm going to guess by the lack of responses and the time that your brains are full. So I think this is a good starting point. Again, you're running out of weekends to study this material. Every weekend is an important weekend, so take advantage of this one. Study hard. I will be releasing your exams right after this. Actually, before I leave the classroom today, I'll do it so that I do not forget for the next 24 hours or so. So tomorrow afternoon, tomorrow late evening, early afternoon, late afternoon, early evening, I will be turning it back off again. So if you're doing the Science Success Center and need to evaluate an exam, this is the perfect opportunity to do that. And of course, you should look over it to see what you did well and see what you struggled with. And as always, if you have questions, feel free to email me. All right. Any questions? All right, then that is everything. Have a great day and a good weekend.